And in today's lecture, we are gonna do the immunology part. In immunology, we'll be starting with some of the basics of immunology, types of immunity and all, barriers and what are the barriers of innate immunity and all. Then go to the acquired immunity and talk about hypersensitivity reactions, talk about complement systems, antibody-antigen reactions, and many things which are there in your microbiology as well as in your biochemistry. So it will cover all the aspects of immunology, nearly all the aspects of immunology which are important from the exam point of view, like for NEET PG and USMLE. Plus it can be watched by both first year and second year students, as well as one who are making basics with starting with the immunology and unit from the medicine as well. And in immunology we'll start. So immunology is basically the study of medicine, and it is derived from the Latin word immunitas. Okay, Latin word is immunitas, which means freedom from disease. So immunology is a branch which deals with the study of the mechanisms which are there in our body for providing resistance to disease. Okay, and it also provides resistance to the cancer development. So it is defined as the system which provides resistance to disease development by microbes, their products, plus resistance to cancer development. Okay, and the person whose immunity are compromised are called as an immunocompromised person. Okay, like for example, HIV/AIDS infected person are immunocompromised, so they suffer from opportunistic infections. Okay, from like Toxoplasma gondii and all infections. Human immune system is basically like an army which contains many cells like NK cells, helper T cells, cytotoxic T cell, macrophages, B cell, plasma cell, T cells, basophils, neutrophils, eosinophils. We'll talk about the cells individually also. Okay, so it is a complete lecture on immunology. Let's discuss the types of immunity. So basically, when we are discussing the types, we have two types majorly. One is the innate immunity, okay, innate immunity, and another is the acquired immunity, also called as the adaptive immunity. Okay, innate immunity is inborn, which we get from the genes, and acquired immunity is acquired during the lifetime. Then innate immunity is present from the birth. Okay, and innate immunity is basically non-specific, like it recognizes pathogens on the basis of their LPS or their gram. Okay, gram positive, negative. In case of gram positive, we have the teichoic acid. Okay, and LPS is present in the gram negative bacterium. You must be knowing this. So they are recognizing broadly pathogens on the basis of some generic molecules. Okay, but these are highly specific. Okay, highly specific, and kill a wide range of pathogens. In innate immunity have a memory stored in that, so memory is stored, and in acquired immunity there is in innate immunity there is no memory. Okay, memory is absent, and in acquired immunity we have the memory present. Okay, the immune cells recognize the pathogens which had infected the host cell earlier, and now a memory is stored due to which they can elicit a higher mode of response, increased magnitude of response when the same pathogen is infecting it. Okay, and immune responses in case of innate immunity, as memory is not stored. Okay, due to this absence of memory, what happens is that the immune response, I'm writing IR, immune response will be of same magnitude and takes same amount of time. Okay, and here the immune response will be of higher magnitude each time, and requires less amount of time because memory is stored. Okay, so this memory is basically the determinant in time and magnitude respect. Okay, let's talk about barriers. What are the barriers present? And barriers are present a part of innate immunity. So we have three barriers, MCB: mechanical, chemical, and biological. What are the mechanical barriers which prevent the microbes? So mechanical barriers are basically like skin and mucous membrane, which are protecting the and preventing the entry of the microbes. So we have the skin and the mucous membrane. I think Chemical barriers, we have the gastric HCl Okay, many things are there, like the pH of stomach. Okay, the gastric acid is responsible for that. pH of vaginal and anal canals and all, many things are there. And biological barriers are basically the commensal bacteria, commensals, the gut commensals which are protecting us, right? Okay, the immune system, in immune system we have something called the first line of defense. So this mechanical barriers form the first line of defense. Skin, mucous membrane, and all the secretions from the skin and mucous membrane, they form the first line of defense. Okay, so form the first line of defense. And second line of defense is formed by cells basically. Okay, so second line of defense is there. Second line of defense is done by some phagocytic cells plus NK cells. Okay, phagocytic cells, you know about macrophages, neutrophils and all. Okay, and we have some antimicrobial proteins and all. Then we have the third line of defense. In third line we have things known as lymphocytes, antibodies, macrophages and all. Done. So adaptive immune response is basically springing to action when there is there is a statement called as the antigen presentation. We'll learn about them in detail. Antigen presentation, I am not discussing it here, but I am just telling you a thing that innate and acquired immunity, this both immunity are not separate entities. They work interdependently. Not independently, interdependently. So basically we have some MSC molecules and all, we'll learn about them, which do antigen presentation. They present the antigen upon the surface, and this antigen is recognized by the adaptive immunity. Okay, just understand this. So this antigen presentation part is done by innate immunity, innate one, and this killing or the action is done by the adaptive immunity. So they are interdependent, right? And there is a question, if do two identical twins have same innate immunity? Yes, because this is present from birth, but they don't have same acquired immunity because it is acquired from the this is acquired after they are born, right? Then we'll spring into innate immunity. Let's discuss about the innate immunity in detail. Innate immunity. So innate immunity will have two components. One is the cellular component, one is the humoral component. So one is the cellular component of innate immunity, one is the humoral component of innate immunity. So cellular component, what we have in the cellular component, we have many cells like neutrophils, eosinophils, basophils, monocytes and macrophages and some NK cells, right? These neutrophils, eosinophils and basophils are known as polymorphonuclear leukocytes, okay, PMNL, right? Or granulocytes, you can say, because granules are present, granulocytes. And in humoral immunity we have, you can remember LIC. LIC stands for lysozyme, which is a bactericidal enzyme present, and present in saliva also, you know. And about interferons, lysozyme, interferons, C-reactive proteins and complements. Two C's are there, C-reactive protein and complements, right? So we'll read about all of this in detail now. Let's talk about neutrophils first. About the cells, you know from your previous knowledge, right? So neutrophils are the cells because they are said neutrophils because they take up neutral stain, neutral or pink color on Jamsa stain. So Jamsa stain, you know, and they take the pink color, like, and these neutrophils are called as the polymorphs or polymorphonuclear cells, polymorphonuclear leukocytes. Okay, they are most abundant of all leukocytes, 50 to 70%, and you must have read this in pathology. If you have read, it is called as the most abundant. I told this one, right? Most abundant. Okay, this is called as the cells of acute inflammation. They are the cells of acute inflammation. and lymphocytes are the cells of chronic inflammation. We'll learn about that. Cells of acute inflammation are the neutrophils. Okay, this is done. And when there is a acute inflammation, these are the first cells which come there. Okay, they act on extracellular bacteria, and these are bacteria non-specific. Why? Because there is a part of innate immunity, right? And this have some receptors also. They have receptors for Fc part of IgG as well as for C3b. C3b is a complement protein. This is a complement. Okay, we'll learn about them, what is their importance later. And these are very good phagocytic cells. You know this, phagocytic cells. These are also known as foot soldiers of immune system. Foot soldiers of immune system. Foot soldiers of immune system. Okay. And neutrophils are basically phagocytic mechanism is there, but for killing they have two mechanisms for killing. For killing they have a non-oxidative mechanism and there is a oxidative mechanism. Let me tell about these mechanisms quickly. So whenever there is a non-oxidative mechanism, what it means is that whenever there is a bacterium, let's say this is a bacterium and this is a neutrophil, so this bacteria will be internalized by the neutrophil. It will form like this and will be taken inside. Whenever there is a whenever it is taken inside, it is called this structure is called as a phagosome, right? And this phagosome fuses with something called as a lysosome. You know this. And this lysosome, when it fuses, it releases some lysosomal enzymes, and there's a formation of phagolysosome, right? I'm writing PL, phagolysosome. It will break the bacteria broken down, and exocytosed. Understood? And okay, so we'll discuss what are the enzymes in lysosomes. So lysosomes have just remember like this, LDH. What is LDH? What are the enzymes? Not LDH. It doesn't have low density. This one. Okay. Not lactate dehydrogenase. Okay. It has lysozymes. It has lactoferrin. This lysozyme will cleave the bacterial cell wall. Okay. This lactoferrin is the iron chelator. So iron chelator, what is the function is that the bacteria or the microbe will require iron for its nutrition. It will chelate the iron. Chelate iron means it will form a complex with the iron, and this complex will decrease the metabolism of the bacteria, right? And this will have defensins. Defensins are cationic protein, and it will cause holes in the bacterial cell membrane, defensins. And we have the hydrolytic enzymes. Hydrolytic enzymes will break down the microbes. Understood? So this is the components of the lysosomes. I told about this. And let's discuss the oxidative mechanism now. Okay, let's discuss the oxidative mechanism. Whenever there is a phagocytosis, there is the microbe is internalized. There is the activation of NADPH oxidase enzyme. NADPH oxidase, and whenever there is activation, there is increased oxygen consumption by the microbe, and this phenomenon is known as oxidative burst. Okay, this phenomenon is called oxidative burst. Okay, so oxidative burst, you understood oxidative burst. And now we are going to so let's if you didn't understood, let me discuss this NADPH oxidase part. So NADPH, what happens is that why this is there in the phagocytic oxidative mechanism and how it kills the bacteria is that this NADPH, in the presence of NADPH oxidase, this enzyme, when it acts on this, it will cause the formation of O2 dot. O2 dot is superoxide anion, and it's a free radical. Okay, and this is the base free radical. This is the principal free radical from which other free radicals like H2O2 and HOCl are formed, and these are all detrimental to the microbes. These are all killing the microbes. Okay, understood. And in case of genetic any deficiencies, that we have a disease which is basically this deficiency of NADPH oxidase. When this enzyme is deficient, we call this as chronic granulomatous disease. Chronic granulomatous disease. So this chronic granulomatous disease is the genetic deficiency which will cause encoding of defective gene for NADPH oxidase. Okay, so this gene for NADPH oxidase is basically present in the sex chromosome, that's X chromosome. Okay, so this chronic granulomatous disease is a X-linked recessive disease. X-linked recessive disease means, you know, this females are the carriers and this males are the diseased. Okay, right? Understood. And this killing capacity of neutrophils is here severely disabled, and we are prone to various infections like from Staphylococcus aureus and all, right? And you must know that neutrophils have a short lifespan, about 6 to 8 hours. And in case of a CGD patient, this neutrophil will not be able to kill, and it will die soon. Okay, die soon. And here in chronic granulomatous disease, what happens is that neutrophils have short lifespan, right? They will die soon. So who will appear at the site of inflammation? Some macrophages will come at inflammation site. Macrophages will arrive at the inflammation site. Macrophages, what will they do? They are very good phagocytic cells, but they too will not be able to kill this type of bacteria. Okay, they are also unable to kill. So they will cause the formation of something called as a granuloma. This macrophages, they are differentiated into, this is a pathological condition, they are differentiated into some slipper like cells which is called as epithelioid cells. This all cells will be unable to kill also. So granuloma is basically an aggregate of cell, right? This aggregation of cell, this when macrophages are coming, epithelioid cells are coming, then there is some aggregation of cells formed, and this aggregation is basically called a giant cell. These giant cells are also many in number. These are also unable to contain the microbe, and they are basically forming the aggregation of various cells which are called the granuloma. And for the diagnosis of this CGD or chronic granulomatous disease, we have something called the NBT test. NBT test is the nitroblue tetrazolium test. Okay, so this is the test. What happens is that when you have the normal neutrophils, we'll add a NBT dye. Okay, this normal neutrophils will cause, when it is added with NBT dye, cause the formation of free radical. Okay, and this NBT dye will accept the electrons from free radicals and get reduced, and when it get reduced, the neutrophils become purple. And neutrophils purple means, you understand, the test is positive and the person is normal or diseased. Yes, person is normal. But whenever this O2 minus is not formed, this O2 free radical is not formed, then in case of CGD, there will be no formation of O2 free radical because there are diseased neutrophils, like neutrophils have died. So there is no formation and test is negative. So we get colorless. So colorless means the person has CGD. Understood NBT test, right? So in CGD patient, no free radicals, colorless, negative CBT test, right? Okay, so these are the things, and Neutrophils, about neutrophils, maybe we have discussed all. Let me repeat you very rapidly. Neutrophils neutral to pink on Jamsa stain, polymorph nuclear leukocytes 50 to 70% in proportion. Cells have acute inflammation, bacteria non-specific. They have receptors for Fc part of IgG. They have C3b complement, receptor for complement system, right? Killing is by two mechanisms: non-oxidative, which is by plain phagocytosis and phagolysosome formation; oxidative by activation of NADPH oxidase, which will cause the oxidative burst, and this ions will be detrimental to the microbes. We discussed about the lysosomal enzymes: LDH, L for lactoferrin and lysozyme, D for defensin, so is insert pores, cationic proteins which insert pores in the bacterial cell membrane. H for hydrolytic enzymes which break down the microbes. Then 6 to 8 hours we have we discussed the CGD, which is the genetic abnormality, X-linked recessive condition in which there is no NADPH oxidase, and NBT test we discussed about that, nitroblue tetrazolium test. The neutrophils means NBT dye. Neutrophils means free radical formation will be there and accept electron and cause purple. Purple means positive, P for purple, P for positive, and P for person is normal, right? And negative means colorless, okay, and person as CGD. Okay, understood. So we discussed all. What do you know? What is a pus? Pus is basically dead bacteria plus dead neutrophils. And this bacteria which induce the formation of pus are called as a pyogenic or suppurative bacteria. Pyogenic or suppurative bacteria. Just remember some examples like Staphylococcus aureus, Pneumococcus. This all, right? So we finished the neutrophils. Let's move on to eosinophils. Okay, so one is done. Let's move on to eosinophils. Eosinophils are called so because they have cytoplasmic granules which have affinity for the acidic dyes. Okay, affinity for acidic dyes like eosin. Okay, and they take a pink color on eosin. Nucleus is basically bilobed. Okay, we have nucleus something like this, or spectacle nucleus. Okay, bilobed or headphone shaped or whatever you can say, this type of nucleus. These are present in the eosinophils, and 2 to 3% in of total circulating leukocytes. Okay, they are weakly phagocytic. They are also phagocytic, but weakly phagocytic. Basically, these are important because they act against the helminths, act against parasites or helminths. They are also non-specific, innate immunity, you know, and they have some granules, some coarse granules are there in the brick red granules. There are brick red granules. Okay, they have brick red granules, and these have basically some components of eosinophilic granules. So we have, first most important is the MBP, that is a major basic protein. This major basic protein is very helpful against these parasites like helminths. Okay, and we have some histaminase enzyme, leukotrienes, and peroxidases. These leukotrienes and peroxidases cause tissue damage and inflammation, inflammatory mediators these are. Okay, and what are the receptors present in eosinophils? So we have basically Fc part of receptor for Fc part of IgE, not IgG. Fc part of IgG which had receptor, your neutrophils, right? Here look at this, Fc part of IgG receptor is present in neutrophils and C3b. Here we have in eosinophils receptor for Fc part of IgE and also for Fc part of IgG. Understood? Both receptors are present. This Ig is basically helpful in the parasitic infections, right? Understood? Okay. So there's a mechanism here. Okay, there's a mechanism which is called as the antibody dependent cell mediated cytotoxicity, or ADCC. Let's discuss about this. ADCC. What is ADCC? Let's understand. So let's say here we have a eosinophil. Here let's say you have a eosinophil. Okay, eosinophil will have granules, and this granules will have MBP. Let's say MBP is there, right? This will have receptor for FC part of IgE, receptor for FC part of IgE, right? Let's say there is a micro parasite here. Let's say this is a parasite. What happens whenever there is a parasite? Okay, whenever there is a parasite, the antibodies will be produced against it, right? This antibody will be bound to it here, like this. Antibody will be, you know about the components, right? FAB part, FC part, two parts are there. So this FC part binds to the FC receptor for FC part of IgE. Whenever there is a binding, there is the release of this granules, exocytosis. MBP will be released, will act on parasite and will kill this. So this is called the antibody dependent, because it is antibody dependent antibodies, cell mediated cytotoxicity, right? Okay, this MBP also has a role in asthma. Okay, this MBP has also a role in asthma because it can damage respiratory epithelium. So it plays a role in asthma. Okay, so we discussed about this eosinophils. Let's revise it very quickly. First thing, affinity for acidic dyes like eosin. Take a pink color, 2 to 3%. Brick red granules are there. A spectacle shaped nucleus or an indented nucleus is present. Not indented word is used in monocytes basically. Spectacular bilobed nucleus, you can say. Weakly phagocytic and act against helminths. It has mainly 4 things which are in the granules. MBP, major basic protein, which has a killing action against the helminths. Mechanism is ADCC, antibody dependent cell mediated cytotoxicity, right? Here the receptor is there for FC part of IgE and FC part of IgG also. MBP histaminase enzyme is present. Leukotrienes and peroxidases are present. Leukotriene peroxidase are basically helping in the inflammatory processes. Okay, tissue damage, they cause tissue damage and healing by fibrosis and all. Many things are there. Chronic inflammation basically. Okay, this eosinophils are also seen in the chronic inflammation. If you have seen in the histopathological sections, it will look something like this. Like half of this will be red and half will be bluish. This type of structure you will find whenever you look at the tissue slides sections, right? Let's discuss next the basophils. Eosinophil is done. Let's move on to the basophils. Okay, basophils, why they are called so? Because they have affinity against basic dyes. Okay, the least abundant, the about 0 to 1%, you can say, or 0.2% in some books it is written. You can follow anyone. Okay, and tissue counterparts, like when these basophils, they move to the tissues. Here in tissues, let's say this is a tissue. They are called as the mast cells. So mast cells are the tissue counterparts of these basophils only, right? And this basophils and mast cells have a role in type 1 hypersensitivity reactions. They have a role in type 1 hypersensitivity reactions. Understood? Then what is the nucleus? Nucleus is again bilobed or horseshoe nucleus, important. Horseshoe nucleus is important, right? Understood? So this much is enough. Then let's discuss about the monocytes. Monocytes. So basically this have 2 to 4% of circulating WBCs. Okay, and when this come to the tissues, they are called as the macrophages. We have a system in our body which is known as the RES. RES stand for reticulo endothelial system. So RES or reticular endothelial system comprise of your circulating monocytes plus tissue macrophages, right? This is the reticular endothelial system. And we have special names of macrophages in different tissues. Okay, this tissue macrophages have special names. Some of the names you must know about. 6 names are there, right? Basically 6 names are there. Microglial cells or jitter cells in brain, just different names but the same macrophages are there, right? And dust cells whenever it is present in the lungs. I'm drawing the lungs, I'm not writing. Kupffer cells, you know this, where KUPFF here. Kupffer cells in liver. Osteoclasts in bone. Mesangial cells in kidney, right? And last is the writing here Langerhans cell in skin. Okay, and just know this macrophages, these are long-lived cells, okay, and have a long life unlike the neutrophils. Okay, then we have something called the CD markers. What are CD markers? You must have, if you have read pathology, you must be knowing that various CDs are there: CD23, CD5, CD40, CD39. Okay, positive in different, these are immunohistochemical markers basically in different diseases. This is a separate thing, but CD molecules are the surface proteins. These are surface proteins on the immune cells. Okay, and the full form is cluster of differentiation. This indicate a lineage of a cell. Okay, more than 350 are there CD markers, and basically the monocytes and macrophage have a CD marker of CD14. CD14 is the marker of monocytes and macrophage, right? Okay. So macrophages, what is their killing mechanism? You must have been knowing the phagocytic mechanism, killing mechanism of macrophages. So you have two types of mechanism. One is the oxidative, one is the non-oxidative. Same thing, reduction of free radical and all. Okay. So monocytes and macrophages are basically active against the intracellular pathogens. Understood? So instead of it, whenever there is any in action by this macrophages or monocytes, there is no formation of pus. Okay, and this type of response in which no formation of pus is there, but there is a formation of granuloma This is called the chronic inflammatory condition, chronic inflammatory response, or granulomatous infection. For example, tuberculosis is the very common granulomatous infection, like leprosy is there. Okay, these things are there. And macrophage understand the receptors. What are the receptors? Let's discuss about that. The receptors on macrophages. So macrophage have receptor for the same Fc part of IgG is there, plus C3b, the same as the neutrophils, right? And macrophages are very important antigen presenting cells, APCs. Antigen presenting cells, just know we have three professional APCs. We'll discuss about this later, but I'm telling you, one is the macrophage, one is the dendritic cells, and one is the B cells, B lymphocytes, right? This is the professional APCs. Very good antigen presenting cells. Understood? So let's discuss how they are presenting the antigen. Okay, we'll discuss the mechanism now, how they are presenting the antigen and which antigen is presented to what cell, right? So let's take it simple. This is a macrophage. Let me tell that this is the bacterium. What will happen? This bacteria will infect this. The bacteria will continue to what it will do? It will try to multiply here, but that it will be broken down by the lysosome. Let's say this is the yellow one, the lysosome. This lysosome will break down this bacteria and it will form some pieces of this bacteria, right? Will form this pieces. This antigen presenting cells, this macrophage in this case, have a receptor for MHC2, have MHC2 molecule present here. MHC2 is basically giving an antigen to it is presenting the antigen here. So here, what is the antigen? These pieces of bacteria are presented here. These are displayed on the surface like this. So it's basically displaying the thing. And whenever there is a display, some people will come to watch, but here the which person comes to kill. Okay, so there is a naive T cell, TH0 or naive T cell. This is called the naive T cell. This comes to picture. Okay, this will see this. It will have a TCR, T cell receptor. It will recognize this. Whenever it recognizes this, it will know that it is a bacteria which has infected it. It will try to kill it, right? There is a confirmatory co-stimulation molecule also here in MHC2. It will touch this part. Okay, we'll discuss about that later. So this T cell have this CD4. This is CD4. We'll discuss about this later. So you understood what is the mechanism happening. So MHC2 is basically having the molecule CD4. CD4 is present on the naive T cell, right? MHC2 is present on the antigen presenting cell. This is present on the TH0 cell. Okay, and this is presenting to the CD4, right? And MHC1 is presenting it antigen to the CD8. PC and the TH CD8. Okay, this is present on the cytotoxic T cells, basically TC cells. So there is a rule of 8 which is followed. This is called the rule of 8. If you didn't understand this part, just go on, we'll understand this. Okay, MHC2 will display exogenous fragment of microbes. Exogenous, okay, because there is an outside infection, right? This is an exogenous microbe. MHC1 will display endogenous fragment of microbes. It will show the endogenous fragment, right? And there's a question, which is the best antigen presenting cell? Whenever someone asks you this best antigen presenting cell, you tell this dendritic cells. These are the best. Understood? These are the mechanisms which are going on. Let me show you something, a picture which will make you understand better. Okay, this part which will make you understand better, and then we'll go to the NK cells via the PPT only. Okay, via the PDF only. Basically, my notes I am showing. Just see the picture first. See this cartoon. There is a protein here. Okay, just see. There's a protein. Let's say this hen is the protein. So it is processed first. It is processed by the proteasome. Proteasome is a cell organelle which is processing the protein. So whenever this protein is processed, it will cause the pieces of this protein. So let's say this leg piece is the piece. Okay, leg piece is there, right? It has caused pieces. These pieces are called as the peptides. So whenever there is a protein, proteasome converts it into peptides, right? And here these peptides are caught by this tap molecules, which is not important for you now, and this is taken by MHC class 1. Okay, MHC class 1 is basically taking it and giving it to CD8 T cell, cytotoxic T cell, and this will in fact kill this one. Okay, it will kill this microbe or this cell, right? This is the whole mechanism. Did you understand by this cartoon? Yes? Okay. Let's move on to next type of cells, called the natural killer cells. Killer cells or null cells are large granular lymphocytes. Okay, LGL. Killer cells, null cells or large granular lymphocytes. Lymphocytes are B cell, T cell and NK cell. NK cell is the innate immunity, and B cell and T cell are of acquired immunity. Just remember this. Cytoplasmic granules have two type of things, perforins and granzymes. Okay, but excellent activity is against tumor cells and virus infected cells. So basically it is effective against TB. What is TB? Tumor cells and virus infected cells. NK cells are not tumor specific or not virus specific. Why they are not specific? Because they are part of innate immunity. What are the markers? CD marker of NK cell? 16 and 56. 16 and 56, right? CD16 because they have receptor part, FC part of IgG antibody. Means these are there, right? So what is the mechanism of killing? What's the mechanism of killing of NK cells? Okay, so NK cells have the same mechanism only. Let me show you. Just focus here. Okay, this part. So let's say this is an NK cells. Okay, and NK cells have granules containing various enzymes. Okay, this is a FC part, receptor for FC part, right? Receptor for FC part is present here, and here let's say we have a tumor cell. This is a tumor cell. This tumor cell will express some tumor proteins here. These are the tumor proteins. Okay, tumor proteins are there, and here that when these tumor proteins are bound by these antibodies. Okay, whenever there is an altered protein, this is a foreign protein, right? Because there is a mutation in gene. Whenever there is a mutation in gene, there is an altered mRNA. Altered mRNA means altered protein. Altered protein means this tumor protein are foreign for the cell. So antibodies will act. This antibody FAB portion will bind to the tumor proteins, but this FC portion will bind to the FC receptor, right, for this cell. Whenever there is a binding, there is the release of this granular contents. These granular contents are released out. This granular contents contain perforins and granzymes. These perforins are basically inserting holes on the tumor cells. This will cause holes. This granzyme will enter. This will enter inside and kill by active apoptosis. Kill by active apoptosis. This is also a mechanism of ADCC, antibody dependent cell mediated cytotoxicity. So ADCC, another mechanism, could you follow, right? This is the whole mechanism which is there in this whole picture, right? I made you understand by this. Okay, So if you understood this, let's move ahead. Erase this. Okay, this is the 4 mechanisms of ADCC. We'll discuss when we're going ahead, right? Cells participating in ADCC, we have 4 cells: macrophage, eosinophil, NK cell, neutrophil. Basophils are not there. Except this, all 4 cells are there. There is no phagocytosis in ADCC. Of course, did you see any phagocytosis in ADCC? No. So ADCC may be a short note for you, important for you. NK cells kill those cells which show down regulation, that is decreased expression of MHC1 proteins. Very important statement. Okay, so these are killing those cells which showing down regulation. Okay, because MHC1 is not there, so NK cells is basically coming to action and killing the cells, right? And antigen presenting cells have both MHC1 and MHC2 on the surface. This point is there. Okay. Yes, mechanism, this type of mechanisms are there. Okay, just a minute. Yeah, let me tell about this lag cells and contact killing mechanisms. Okay, just focus. Let's say this is a normal cell. This normal cell has a class 1 MHC molecule. Just understand very carefully what I am saying. This normal cell have a class 1 MHC molecule present on the surface. This is present. With this we have a AR ligand. Okay, we have a AR ligand which is attached to this normal cell. And this NK cell have a inhibition receptor and a activation receptor. 2 receptors are there. There is a inhibition receptor which will bind to the class 1 MHC. When this bind to the class 1 MHC, they give a negative feedback to the nucleus. They give a negative signal to the nucleus. This is signal transduction mechanism. So negative signal to the nucleus. And whenever the activation receptor and the AR ligand, whenever there is a protein fragment between forming a bridge between AR ligand and activation receptor, this one, so it is giving a positive feedback. Okay, whenever there is a negative and positive feedback, they are nullifying and there is no killing. So normal cell is not killed. We don't want normal cells to be killed, right? Okay, but let's say we have a virus infected cell. So in virus infected cell, what I told earlier, look at this. NK cells kill only those cells which show down regulation, that is decreased expression of MHC1 molecule. Very important statement. So there is a down regulation of MHC1. So basically, whenever there is a virus infected cell, the MHC1 is not expressed. So this ARL is there, activation receptor is there, protein fragment is also there. This is normal, positive signal is there. But this inhibition receptor is not able to bind to the MHC1, right? So there is no negative feedback. Whenever there is no negative feedback, what happens? It is activated and will cause the release of perforins and granzymes. This perforins and granzymes will kill the virus infected cell. Yeah, understood? This is the whole mechanism of killing of virus infected cells by NK cell. Understood? If you did not understood, let me make you understand again. We have a normal cell here. This is the normal cell. Let me write N. So normal cell will have MHC1, and there is a AR. Okay, there is a AR also. AR ligand. Sorry. Yeah, what I am saying. Okay, and there is a NK cell. So NK cell will have. Let me write here. NK cell will have a nucleus. And NK cell will have a activation receptor and inhibition receptor. This is the inhibition receptor, this is the activation receptor, this is the protein fragment, which is basically acting as ligand. Whenever there is a interaction between the inhibition receptor and MHC1, there is a negative feedback going inside, and this will also only give positive feedback. Positive negative is nullifying, and nullify means no killing. But what happens in tumor cells? In this tumor cells, this MHC1 is not expressed. This is not there. So inhibition receptor is only there, which is free. This is free, and whenever it is free, it is not giving an positive or negative signal, it is giving null signal. Basically one positive signal is received by this, and one positive signal means there is no nullification and there is no killing. There is a killing by what? Perforin synchro enzymes, they are killing this cell. Yeah. Understood? Clear? Crystal clear, right? Okay. Let's see. Another thing is that the inactivated NK cell, so interleukin 2 and 12, just remember 212, interleukin 2 and 12 are basically activating NK cell. This activated NK cell is called as the lymphocyte activated killer cell, because lymphokine activated killer cell, okay, not lymphocyte, these are lymphokines, right? Cytokines, okay, lymphokines activated killer cells, LAK cells. Understood? LAK cells. Okay. Another type of killing is called the contact killing by NK cells. Contact killing means, what is the mechanism? Whenever there is a NK cell and there is a target cell, this target cell, whenever there is a Fas protein bound to the target cell, Fas protein is the death protein. This Fas protein is recognized by the Fas ligand, FASL. Okay, this is also written like this, FASL, FASL. Okay, Fas ligand. This Fas ligand is basically recognizing this Fas protein, and there is an interaction, and whenever there is an interaction, there is a killing. Okay, this NK cell will kill this one, death protein. Understood? So this completes your NK cells and all the cellular components. We'll move to the humoral components the next, right? Let's move on to the humoral component of innate immunity. So we have what we have: LIC, I told you, lysozyme, interferon, CRP, and complement, right? LIC. So we're discussing the LIC now. Lysozymes. What are lysozymes? This can be a short note, just pay attention. Was discovered by Sir Alexander Fleming. This was Sir Alexander Fleming who discovered lysozymes. It is present in all body secretions, except CUS, except CUS. CUS means CSF, urine, sweat. Lysozyme is absent in CSF, urine, and sweat. Antibacterial action, it is damaging the bacterial cell wall or peptidoglycan cell wall, and it is bacteria non-specific, which is part of innate immunity, right? Okay. After this, let's move on to the C-reactive protein. Or CRP. Here C stands for carbohydrate reactive protein. So liver is producing the CRP, and the CRP is released in the circulation. Understood? Whenever there is a organism like pneumococcus or streptococcus pneumoniae containing organism, there is a carbohydrate which is attached to the cell membrane. Okay, the carbohydrate is attached to the cell membrane of this pneumococcus or streptococcus pneumoniae. Okay, and the C-reactive protein is basically reacting with this part. Okay, so it was named as CRP. Understood? CRP is not an antibody. Okay, it is not an antibody. It is just that the reaction occurred, so it was named as C-reactive protein. Antibodies are gamma globulins, but CRP is a beta globulin protein. Understand? Beta globulin protein. Levels of CRP increased in any type of acute inflammation. CRP is also known as acute phase reactant, ARP. Okay, APR, acute phase reactants. Understood? CRP is a non-specific marker of acute inflammation. It is used to monitor the activity of autoimmune disorder. It's a prognostic marker, very important. So in rheumatoid arthritis and SLE, DLE, and other disorders, which are autoimmune disorders, this SLE, basically this CRP is a marker of acute inflammation in this type of disorders. And CRP levels are assessed by agglutination reaction and precipitation reaction. Two reactions are there which are used to assay the CRP levels. Assay means quantifying the levels of CRP. Okay, and among the agglutination reactions, we have the latex agglutination, which is commonly used. Okay, what happens is that we have the latex particles like this, latex particles which are coated with the antibody. They are coated with the antibody like that. Here, antibody coated. This antibody coated latex. Okay, what I'm saying. Particles are added with the analyte. Okay, what we have to analyze. So whenever there is an analyte, there is a cross linking and clumping. There is a clumping, and this clumping is assessed. Okay, So CRP levels are assessed by agglutination and precipitation reactions. After CRP, what we learned about CRP? So CRP is a C-reactive protein. It's a carbohydrate. Then it's produced by liver. It's released in circulation. It recognizes the carbohydrate chain of this pneumococcus and streptococcus pneumoniae, which is binding to it. Hence, it was named CRP. CRP is a not a beta globulin, gamma globulin. It is a beta globulin, right? It is a beta globulin. And levels of CRP is a marker of acute inflammation. So it is an acute phase reactant, also known as APR or acute phase reactant. It's a non-specific marker of acute inflammation. Okay, it's a prognostic marker also, means autoimmune disorders are quantified and diagnosed here. Okay, prognostic marker, prognosis. So rheumatoid arthritis and SLE, both in this CRP levels are measured. CRP levels are assessed by agglutination reaction and precipitation reaction. Two reactions are there. Agglutination reaction, the latex agglutination is commonly used. Here the latex particles are coated by antibody, and when an analyte is given, there is an agglutination. A precipitation reaction is there also. Interferons, you know about basically these are okay. Interferons we haven't discussed, right? I have to, yeah, no problem. Let's discuss the interferons now. Okay. Let's talk about interferons. Interferons can also be a short note question for you. Interferons are basically glycoproteins. Remember, it is a glycoproteins. Okay, glycoproteins. And we have type 1 interferons and type 2 interferons. Type 1 means alpha and beta. Type 2 is interferon gamma, right? Type 1 and type 1 are antiviral interferons, okay, against virus. Alpha and beta are antiviral, interfere with the activity of viruses like viral translation. They interfere with viral translation, right? And this interferon gamma, which is an immune interferon, this is produced by Th1 cells and NK cells. Th1 cells is a subset of T cell. Okay, we'll discuss about that. So interferon gamma is not strictly a part of innate immunity. It's not strictly a part of innate immunity. Okay, because it is produced by Th1 cells. Th1 cells means T cell. T cell means cell mediated immunity, right? Okay, and this viral interferons are viral non-specific. So about interferons, what you learned? Interferons we have two types. We have the type 1, okay, type 1 interferons. We have the type 2 interferons, and this type 1 we have IFN gamma, sorry, alpha, and IFN beta. Here we have the IFN gamma. These both are antiviral and these are non-specific, but this interferon gamma is produced by Th1 cells and the your NK cells. Okay, and this Th1 cells, because it is produced by Th1 cells, not strictly innate immunity. Understood? Okay. After you understand this classification, let's move ahead. Okay. Yeah, so now let's discuss the mechanism of interferons. Okay, let's discuss the mechanism. Whenever there is a viral infection, how the cell is killed, how the virus as well as how the cell is killed, because both virus and the cell will be killed here. Okay, so we have what will this form, let me tell you, then we'll discuss the mechanism in the fresh phase also, because these mechanisms are very important. You must have to understand, okay, very carefully. Let me first teach you by this PDF, then I will discuss the mechanism in a fresh phase also. Okay. So whenever there is a viral infection, let's say this is a virus entering here, there is the production of interferons. Okay, there is the production of interferons. This is the host cell 1. This is the host cell 1. Let's say this is infected by virus, interferons are produced. These interferons bind to this receptor for interferon, which is present in the host cell 2, right? Host cell 2. Host cell 2 has 3 antiviral proteins. Each cell has 3 antiviral proteins, 3 type of antiviral proteins. Okay, the red one, blue one, this purple one, right? And it will get activated against any viral infection in future. 3 viral proteins are the 1, 2, 3, right? So this is uninfected cell. So if this is infected, it will look like this. This is the virus, and this is the host cell 2, right? Yes, and this is the virus. This is the 1, 2, and 3. Let's zoom into the cytoplasm. What are these viral proteins and what they will do? First viral protein is called the inactive oligoasynthetase. Inactive oligoasynthetase. Okay, understood? Inactive oligoasynthetase. This inactive oligoasynthetase, what it will do? It will activate the dsDNA. Okay, formed during the viral replication. Okay, this inactive oligoasynthetase will be activated by that dsDNA, which is formed during the viral replication. When the virus will replicate, it will form the dsDNA, activate the dsDNA, and it will form the active oligoasynthetase. This active oligoasynthetase will produce some protein known as oligo A. Okay, and this oligo A protein will activate again another protein which is numbered as 2 here, this blue protein. Okay, it will activate the blue protein. This is called the inactive ribonuclease. This inactive ribonuclease is activated by oligo A. This inactive ribonuclease, when it's activated by oligo A, it will form the active ribonuclease. Okay, active ribonuclease. It has both viral and cellular mRNA. Active ribonuclease. And again, this inactive protein kinase, which is labeled as 3, inactive protein kinase is activated by this dsRNA produced during viral replication. This dsRNA produced during viral replication will activate the inactive protein kinase. Okay, this is active now. This will stimulate the binding, the phosphorylation of elongation factor 2. Elongation factor 2 you must have read in the molecular basis of inheritance, okay, and biochemistry. When there is a phosphorylation of elongation factor 2, is inactivated. So EF2 is phosphorylated and it gets inactivated, so there is no protein synthesis. Whenever there is no protein synthesis, there is cell death. Host cell 2 will die, right? What happened? Did you understand the mechanism, what is happening? But viruses along with will also die because virus is inside that, right? Could you understand the whole phenomenon, what is occurring? Okay. If you want me to discuss this again, I am taking a separate page. Let me first finish the interference, then we'll discuss the mechanism again. Okay, because this is an important mechanism I want to discuss with you again. Okay, yeah, we'll discuss the mechanism again. We'll come back to this. Interferons are virus non-specific but species specific. These are species specific. Interference of humans will act on humans only and not on monkeys and all. Okay. Interferons are produced early in immune response in case of viral infection, before the antiviral antibodies are present. Antiviral antibodies are take time actually to produce. Okay, they are very late to be produced. So interferons are basically providing a fast response. Okay. Interferon alpha is produced by leukocytes. Interferon alpha is produced by leukocytes. You can look this alpha, and alpha means L type of thing. And beta is produced by fibroblasts. Very good source of antiviral interferons are a group of plasmacytoid dendritic cells, PDCs. Okay, we have some cells called PDCs which form the antiviral interferons. Okay, alpha and beta. Main production parts are the leukocytes and fibroblasts, but plasmacytoid dendritic cells also may produce the antiviral interferons. Okay, those are different from the normal dendritic cells. Okay, here let me take some space and discuss the mechanism of interferon action. Okay, mechanism of interferon action. So let's say this is the host cell 1. I am writing HC1. Okay, this is SC1. Let's say here is a virus which is coming here. This is infecting it. This virus infection will cause the formation of interferons. This will act on a receptor for the interferons R for interferons. It will act on the receptor for interferons and will activate 3 enzymes. So we have 1. Same color. I will take 2 and 3. 3 enzymes are there. These are antiviral proteins. Okay, 3 not enzymes. You can say protein or enzyme. You can say these are antiviral proteins. 3 antiviral proteins are there. Okay, and this is the host cell 2. C2 I am writing. this is infected. Now interferons will bind, it will cause the stimulation of this part. So first one is the inactive oligo A synthesis. Inactive, first one I am talking about, first one inactive oligo A synthesis. This inactive oligo A synthesis is activated by dsRNA, which is formed during viral replication. The formation of dsRNA. This dsRNA will activate this. Active, will form the active oligo A synthesis. This will cause the synthesis of oligo A protein. This oligo A protein will cause the activation of another enzyme, which is called the inactive ribonuclease. The second protein, inactive ribonuclease. This inactive ribonuclease is now activated now due to the presence of oligo A. This is activated now, and this another third protein is the inactive protein kinase. Inactive protein kinase is activated again by the dsRNA during the viral replication. It is now forming the active protein kinase. Is active protein kinase, what it will do, it will cause the phosphorylation of elongation factor 2. This phosphate group is attached to the elongation factor 2, phosphorylation, no protein synthesis, and whenever there is no protein synthesis, there is cell death and killing. Okay, cell death and killing. Could you understand the whole mechanism now? I repeated the whole mechanism 2 times, and I hope you understood this all. Okay, let's move on to the next part now. Let's discuss about the components of acquired immunity. So we have some cellular components as well as humoral components. As in innate immunity, we had some cellular and humoral components, right? All the cells we discussed, and humoral components we discussed about LIC, you know, lysozyme and interferons and complement and C-reactive protein. Components of acquired immunity, we have cellular components and humoral. Cellular is the T lymphocytes, so T cells, and CD marker of T cells is CD3, very important, please remember it. Okay, and humoral immunity, acquired immunity, humoral component, we have the antibodies produced by the plasma cells, right? So these T lymphocytes are of basically 2 types, helper T cells and cytotoxic T cells. Many classifications are there, we'll discuss in due course of time. So helper T cells, the pan T cell marker is CD3 will be positive. Helper T cells will have CD4 positive, cytotoxic will have CD8 positive, right? And HIB is main receptor is CD4, so it affects the TH cells and also affects the macrophages and monocytes, because CD4 is also present on macrophages and monocytes. Understood? And let's discuss about the CD markers of this antibody, the B cells. So B cells are producing the antibodies. Fully differentiated B cells are called the plasma cells. These plasma cells, when they are fully differentiated, produce the antibodies. One plasma cell can manufacture 2000 antibody per second. Golgi apparatus is very well developed in plasma cells. CD marker B cells are CD19, 20, 21, 81. Okay, 19, 20, 21, 81. Understood? Let's discuss the difference between T lymphocytes and B lymphocytes. T lymphocytes are called so because they mature in the thymus. B lymphocytes are called so because they mature in the bursa of Fabricius in birds, which is equivalent to the bone marrow in humans. You can see here, it's a chicken where there is a bursa fabricius present here. Okay, and this is equivalent to the bone marrow in case of humans, so it was named B lymphocyte in that way. Okay, T lymphocytes are formed in the bone marrow. Bone marrow produces the immature T cells and they go to the thymus for maturity. Okay, these are the things, and you can say CD3 positive T cells are CD3 positive obviously, and this B lymphocytes are CD19, 20, 21, 81 positive, right? And both T cells and B cells recognize specific antigens, okay, these are acquired immunity is specific, and unique antigens also. So TCR and BCR, we have structures of TCR and BCR which has to be discussed. Okay, TCR is basically a heterodimer receptor. Okay, TCR is a heterodimer receptor. We have a alpha subunit here, we have a beta subunit here, we have a constant region of beta, and we have a variable region of beta, BC and BV. Okay, and likewise we have BC, BV and BV, AV. Okay, beta C, alpha C, that's the constant parts. Alpha will have the same subunits, right? Beta C, alpha C, okay, and beta V, alpha V, right? One TCR binds to a single protein antigen in the pocket formed by alpha V and beta V region. Okay, just let me show you, just make you draw a structure. If this is a T cell, okay, let's say this is a T cell, we have the alpha, this is the alpha b, this is the alpha c, right? Sorry, alpha c, and this is the alpha b, right? And this is the beta c, constant region of beta, this is a beta b. So antigen, the this part will be bound here, this antigenic peptide, the single protein antigen will be formed a group between beta b and alpha b. So this is the antigenic peptide which is formed in the group between alpha b and beta b. Understood? Clear? Yeah. Okay. And another thing, T cell binds to only those protein antigens which are brought to them by MHC molecules. They don't bind to free antigens. Very important. T cells don't bind to free antigens at all. They bind to those which are brought by the MHC molecules. Understood? Okay. And we have its TCR CD3 complex. So T cell receptor, TCR, basically when we say TCR, T cell receptor, it will contain a TCR plus CD3. Okay, this is a TCR complex. TCR complex consists of TCR and CD3. Understood? And CD3 is a signal transduction complex, signal transduction molecular complex, you can say. Okay. This is the whole structure of the TCR CD3 complex. So if you want to see, this is the whole structure. Alpha b, beta b is there. Okay, and this is the whole TCR structure which I told you now. Alpha c and beta c, you know, and CD3 which have epsilon and delta units. Here CD3 will have gamma and epsilon units. Okay, this epsilon unit is same here, epsilon unit is same here. This is delta and this is gamma. Okay, this subunit is different in this both CD3s. Okay, and we have a zeta chain. We have a zeta chain. Understood? This is the whole concept, and it cannot bind to free antigens, so we know this. Okay. And another thing is the B cell receptor, BCR, which is called the BCR. So we have two BCRs, on the first BCR and the second BCR. Okay, first BCR is for IgM binding and second BCR is for IgD binding. Okay, so BCR also known as MIG or SIG, this is membranous immunoglobulin or surface immunoglobulin. Okay, when we tell this BCR complex, likewise we had the TCR complex. TCR complex had TCR and CD3. So BCR complex will have one BCR for IgD, one BCR for IgM, right? M and D we wrote here, yeah. And another one is the Ig alpha, Ig beta, Ig alpha, Ig beta. Okay, then CD19, 20 and 81, CD19, 20 and 81. Okay, this whole comprise of the BCR complex. Understood? So one BCR can bind to two identical antigens, which can be protein, carbohydrate or nucleic acid, because there are two BCRs, right? Two BCRs are there, so it can bind to two which are similar, two identical antigens. The important thing is that binds to two identical antigens. Okay, and can bind to free antigens. It can bind to free antigens, doesn't need any antigenic presentation or MHC presentation. Understood? And activators, what are the activators of T cell? T cell is activated by phytohemagglutinin, okay, phytohemagglutinin and concavalin A. Concavalin A is a toxin. B cell is activated by Epstein-Barr virus and LPS of gram-negative bacteria. LPS is present in the gram-negative bacteria, right? Okay, and Epstein-Barr virus, EBV. And phytohemagglutinin and concavalin A are the activators of T cell. So these things you must remember. BCR and TCR structures you should know. BCR complex, again I am repeating, you will draw a cell like this. Okay, and if you want to draw in exam, right, you have to draw in exam, right, obviously. And if you want to see the structure again, let me tell you, this is the thing. And we have two BCRs here. Here is one BCR, here is one BCR. Okay, one for IgM, one for IgD. This is one fact. And one is Ig alpha and Ig beta. We'll draw Ig alpha, Ig beta here. You will draw Ig alpha, Ig beta here. And then three markers you will write: CD19, CD20, CD81. Done. So this is the structure. After discussing the structure, we are going to the next PDF. And we'll start about we'll read more about the acquired immunity. Let's move on to the next part. Let me tell you about antigen. What is an antigen? So we'll read about antigen in detail and then we'll move to the MHC complexes. So anti means antibody, gen means generator. So they are generators of antibody. Antigen means they are generators of antibody. So production of antibody is an example of immune response. Okay, antibody production is an example of immune response. And antigen is a foreign substance which enters the body, stimulate the immune responses. So foreign substance stimulating the immune responses are called as an antigen. Antigen is a foreign substance which stimulates the immune responses. A good antigen has two properties, immunogenicity and antigenicity. Okay, you can remember by mnemonic AI, antigen. Okay, antigen, right? AI, antigenicity, immunogenicity. Immunogenicity means it has an ability to induce the immune response. Antigenicity means it's a property to bind to the antibody. Okay, so antigen has a specific site which is called the epitope. So epitope is the site where the antibody will bind. So epitope is the smallest part of antigen which has the property of antigenicity and immunogenicity. The smallest possible part which has both the properties of antigenicity as well as immunogenicity is called as an epitope. Okay, and the smallest antigenic determinant is defined as the smallest antigenic determinant is epitope. Very important. Okay, so here is the epitope, you can see, and here's the paratope. This paratope is present in the antibody. Okay, paratope is the binding site of the antibody with the antigen. So this epitope and paratope bind to each other. So epitope is present in the antigen and paratope is present in the antibody. Understood? Paratope binds with the epitope. And antigen has multiple epitopes. Important. Antigen has multiple epitopes. It has multiple epitopes and it will bind to the corresponding antibody only. That is specificity of antigen antibody reaction. But each epitope, it will bind specifically. It has multiple epitopes, one for each. Okay, it has multiple epitopes, one for each. Sometimes this specificity also fails. There are many cases, diseases in which specificity also can fail. Okay, like let's say here you have an antibody 1 and here you have antibody 2. So antibody 1, here is the paratope and this part is the paratope. This beaker shaped part is the paratope. This one beaker shaped part and this test tube shaped part. Okay, this part and this part are paratopes. And here also we have this same test tube shaped part and another flask shaped part, let's say. Okay, this thing we have, and we have antigen 1 a circle and a triangle, and antigen 2 a circle and a square, right? So antigen 1 must bind here and antigen 2 can bind here. Okay, but what happens, this part and this part are same. Okay, so anything can bind here. This part will bind to here and this part can also bind to here. Okay, the specificity, as it is similar, this can cross bind. Okay, sometimes this specificity fail. This failure of specificity can lead to many disorders like acute rheumatic fever. Okay, this acute rheumatic fever is the failure of specificity of binding of antigen and antibody. Okay, so acute rheumatic fever is caused by the organism group A Streptococcus pyogenes. Okay, group A Streptococcus pyogenes, where M protein, also similar to human antigen, means there is a property of molecular mimicry. Molecular mimicry means there is a M protein which is similar to human antigens. So it gets bound means where there's some binding of human antigen, where M protein gets bound and the body thinks that it is a human antigen, so it elicits a autoimmune reaction. So acute rheumatic fever is an example of autoimmune disease. But the phenomenon is your molecular mimicry, phenomenon of molecular mimicry. Let's discuss about incomplete antigen or hapten or pro-antigen. Okay, what is a pro-antigen? So usually low molecular weight molecule which has got the property of antigenicity but not of immunogenicity. Okay, so where the antigenicity is present but there is no immunogenicity, we call it as hapten, and this hapten is also called an incomplete antigen. So this is the concept of hapten. Okay, and we can change a hapten to a complete antigen by combining with another protein carrier molecule. So the hapten plus a protein carrier will cause a form a complete antigen, right? A hapten plus a protein carrier will form a complete antigen. What are the scenarios? Let's say here is a hapten H. Okay, this hapten is in first time injected. Okay, there's no primary immune response. So say hapten can alone induce a immune response? False. A hapten cannot alone induce a primary immune response. A hapten cannot induce a primary immune response alone. Okay, when a hapten plus a protein carrier, that is forming an antigen, is introduced, there is a primary immune response. Primary immune response. Okay, because now it is a complete antigen. But alone hapten is not a complete antigen. It will not produce an immune reaction. The hapten plus protein carrier, okay, can elicit a primary immune response where IgM will be there against the hapten, IgM will be produced against the protein carrier, and IgM will be produced against the hapten plus protein carrier. So there are three, basically production of three parts. Okay, three scenarios are there. Okay, antibodies to hapten will be formed now, because hapten carrier conjugate this one. Okay, means this is a complete antibody, complete antigen, sorry. So when it was immunized in rabbit first time, so antibodies against hapten were produced, antibodies against carrier molecule were produced, and antibodies against hapten carrier conjugate were produced, right? But when there is a second exposure of hapten, where hapten is exposed for the second time, there is no secondary immune response. Okay, because hapten lacks immunogenicity, it cannot induce the formation of immune response. Okay, hapten doesn't have immunogenicity. No, it has antigenicity but cannot have immunogenicity. But when hapten plus protein is injected second time, there is secondary immune response, a high yield immune response, a secondary immune response which is more exaggerated. The primary, then this will be a secondary. So IgG against H hapten will be formed, IgG against P, and IgG against H plus P. First this was from IgM. IgM is the primary one, and IgG the secondary immune response. IgG will act, right? Understood? So this is the concept of hapten. Okay, so four scenarios are there. One is the only hapten, no immune response. Okay, no primary immune response. Then hapten plus protein carrier will introduce, where there is a primary immune response. Okay, hapten alone was second exposure, there will be no primary immune response, and hapten plus protein second exposure will cause the secondary immune response. Okay, so these are the scenarios. Understood? Yes. After that, we're discussing the determinants of the strong antigen. What is a strong antigen? Strong antigen is the one antigen which can generate a high titer of antibody. High titer means high concentration of antibody. So one which can generate a high concentration of antibodies are called as a strong antigen. Okay, strong antigen. So what are the factors which are determining the strength of an antigen? Okay, what are the factors which are determining the strength of an antigen? Okay, so we have some factors which are there. You can remember by CSDS. Okay, you can remember by determinants of strong antigen. You can remember CSDS. Okay, CSDS. C for chemical nature. Okay, C for chemical nature. First thing, chemical nature means proteins are excellent antigens, followed by carbohydrates, followed by lipids, followed by nucleic acids. Proteins are excellent antigens. Okay, mostly all the antigens are proteins itself. Okay, so proteins, carbohydrates, lipids, nucleic acids. Okay, understood? PCLN. Understood? So this is the thing. Then size of antigen. So C is done. S is for size of antigen. Very big or very small are poor antigens. Mid-sized molecules, obviously anything which is mid-sized is stable, right? So mid-sized molecules are the best antigens. Remember this. Then D for degree of foreignness. Degree of foreignness. How much foreign is that? More the foreign the molecule is from us, better antigen is. Obviously, if one human, if from any antigen is injected from human, the antibody produced will be very less. Human to another human, human 2 to human 1, very less. But if it is an animal, okay, if an antigen comes from an animal, there is a high titer of antibody will be produced, a high titer will be produced, and this shows us that the degree of foreignness matters very much. More the foreignness, more is the production of antigens. Okay, and then we have susceptibility to tissue enzymes. How much susceptible it is to tissue enzymes? Okay, so one antigen which is fully susceptible to tissue enzyme, fully susceptible, there will be very small antigens will be formed. Okay, very small antigens which will not be recognized again, and immune response will be less. Okay, so bad antigen, so very small antigens will be formed and less immune response will be there. But let's say this is the antigen and it's a very good antigen. Okay, it is a very good antigen, and it is why it is a good antigen, because it's partly susceptible. It is partly susceptible to tissue enzymes. Susceptibility means if it is an antigen, okay, if there is highly susceptible, the tissue enzymes will be able to break it into very small parts. There will be only very small fragments which will be not be generating the any immune response. Okay, but if let's say this is the antigen and the tissue enzymes are breaking it in this way, very less susceptibility. Okay, so this type of things are formed. But just think, if this part is foreign, so this part is foreign and this part is foreign and this side parts are foreign, but this parts are also foreign, right? Because it is a segment. This part is different, this part is different, this part is unique, this part is also unique. So you can see there is the uniqueness increases. Uniqueness increases means what? Uniqueness increases means 3 mid-side antigens will produce, and these parts are unique now. This body hasn't been exposed to these parts, means there are newer epitopes exposed. Very important. Are you understanding? There are formation of newer epitopes. The formation of newer epitopes. That means that there will be an increased immune response. There will be an increased immune response. Understood? So more the enzyme is less susceptible, that is partly susceptible. If it is partly susceptible to tissue enzymes, there will be newer exposed of newer epitopes and good antigen. Understood? Okay, let's discuss about the specificities of an antigen. What are the specificities of an antigen? So we have basically 5 types of susceptibility. Okay, we have first species specificity, organ specificity, and heterophile specificity, individual specificity, and isospecificity. Okay, so we have 5 types of specificities. What are they? Let's discuss. Okay, so antigen is basically species specific. First thing is the species specificity. Just a minute. Yeah. Species specificity. The antigens are present in all members of a species exclusively and not in any other species. Antigens which are present in all members of a species are species specific. That is, that antigen of the same species, that antigen will be present in another individual of the same species is same. Okay, not exclusively and not in any other species, right? Then we have organ specificity. So antigens found specifically in an organ is not in present in species, like brain tissue antigen of human and mouse. Okay, the brain tissue antigen of human and mouse are nearly same. Okay, but when the neural rabies vaccine was discovered and it was developed, it was initially made in mouse brain. Okay, but it caused neuroparalytic complications, so it was banned. This is an example of neural. Okay, so antigens found specifically in an organ and not in species. Antigen found specifically in an organ and not in species. Heterophile specificity means different lab. Different lab means what? Is that some antigens are present in different species and not organ specific, but has affinity for each other. Some antigens tend present in different species. Let's say present in human, present dog, and present in cat. Not organ specific also. They are not present in same organ also. Okay, let's say human skin, dog pancreas, cat intestine, but have affinity for each other, means they are nearly same. Here human skin antigen is same with dog pancreas antigen, is same with cat intestine antigen. Okay, this is an example, just same antigen. What are the real examples? This is the you can say this type of example which we give commonly for making you understand. But we have some examples like Will-Felix reaction. Will-Felix reaction, it is used in a tentative diagnosis of rickettsial infections. Okay, and we have cold agglutination test and Streptococcus MG agglutination test. Okay, here tentative diagnosis of Streptococcus pneumoniae. These all reactions are based upon this heterophile specificity. Understood? Okay, then we have individual specificity. What is individual specificity? Antigen uniquely present in one individual of a species and not in any other individual. this antigen is uniquely present in one individual, not in any other individual. Okay, so all of us are antigenically unique except identical twins. Identical twins are antigenetically same. Okay. Examples are the MHC antigens, the genes present in chromosome 6p, so term chromosome 6. Understood? Okay. Then we have isospecificity, that is variations of an antigenic group distributed in members of a species. Variation of an antigenic group distributed in members of a species, ABO blood group and Rh antigen. Okay, means one antigenic group varies and distributed in different members of a species. Understood? Like 85% population Rh positive, rest is Rh negative. Okay, so ABO and Rh are examples of isospecificity. Examples of individual specificity is your MHC antigen. Antigen is uniquely present in one individual and not in any other individual. And heterophile specificity, we have Will-Felix reaction, cold agglutination, Streptococcus MG agglutination test. Will-Felix is for rickettsial infection. This cold agglutination, Streptococcus agglutination is for Mycobacterium that pneumonia. Okay. Organ specificity is for that example of neural rabies vaccine, you can write this, and brain tissue antigen of human and mouse, you can give this. And species specificity are, you understood, antigens present in all members of a species exclusively and not in any other species, right? So this completes an antigen. Let's move on to the next part of immunity. Now let's talk about major histocompatibility complex. So basically, it is a group of genes encoding proteins which can bind to peptide fragment and present them for recognition to the T cells. Okay, it's a group of genes encoding proteins which can bind to peptide fragments. Present them for recognition to T cells. Right? Very simple definition. So MHC genes are present on almost all human cells, but they were first discovered on the surface of leukocytes. Okay, so it was known as human leukocyte antigen. First time discovered. That's why. Not any other reason. Responsible for unique identification of different members of a species. Okay, unique identification of different members of a species, they are responsible for that. Understood? And they are involved in graft acceptance and rejection. They are involved in graft acceptance or graft rejection. Responsible for unique identification of different membrane of a species. In humans, MHC genes are present of chromosome 6p, p arm of your 6th chromosome, right? We have MHC1, MHC2 and MHC3. MHC1, MHC2 and MHC3. MHC3 has other functions. We'll discuss about that later. Okay, MHC1 have 2000 kilobase pair long. MHC2 is 1000 kilobase pair long only. 2000, this is 2000 and this is only 1000. Okay, MHC1 has 3 classes of loci of genes. HLA-A, HLA-B and HLA-C. Okay, HLA-A, B and C. And MHC2 has three regions, your DP, DQ and DR. DP, DQ and DR. Okay, it has 3 regions. Here HLA A, B and C are 3 different entities. HLA A has an alpha chain, okay, and beta 2 microglobulin. This beta 2 microglobulin, the gene is present in 15th chromosome. Just remember this. Okay, and HLA B has a different alpha chain, and the same beta 2 microglobulin, and HLA C will have another different alpha chain other than this HLA A and B and a beta 2 microglobulin. So this beta 2 microglobulin gene is present on chromosome number 15, right? MHC1 proteins are expressed on the surface of all nucleated cells. Present on the surface of all nucleated cells except sperm cells and platelets. They are absent are platelets, not present on platelets and sperm cells. Okay. MHC1 in RBC is present protein antigens to the TC cells. Okay. You know these things. Okay, in presentation I discussed about this previously, right? So we discussed about MHC1, we discussed about MHC2. So MHC1, you understood that it is 2000 kilobase per long, this 1000 kilobase per long. It has 3 classes of loci of genes. You have HLA A, B and C. Here we have 3 regions, which we have DP, DQ and DR, DP, DQ and DR. Okay, this HLA has a alpha chain and a beta 2 microglobulin. This is a alpha chain beta 2 microglobulin, and this beta 2 microglobulin is the chromosome number 15. The gene is present on chromosome number 15. We discuss all this. Okay, and where this is present, MHC1 is present on all nucleated cells except your platelets. Platelets and sperm cell doesn't have this MHC1 molecule. Understood? This much is clear. Let's move ahead. Okay, so let's discuss about MHC2. MHC2 has three regions: DP, DQ, and DR. Three regions, you can say, DP, DQ, and DR. You can these three regions act as three different MHC2 molecules. There are three different MHC2 molecules, in fact. Okay, and all alphas and betas are entirely different from each other. So DP has alpha beta, DQ has also alpha beta, and DR has also alpha beta. So alpha beta is present in all, but this alpha is different from all of this. This beta is different from all of this. So DP, all alpha and beta are different. This DQ, alpha and beta are different. This DR also, alpha and beta are different. So all these entities are different. Three different MHC2 molecules. Okay, all these entities are different. All alphas and betas are entirely different from each other. All alphas and betas are entirely different from each other. Understood? Okay, so class 1 and class 2, this is the structure. Alpha 1, this is alpha 1, alpha 2, alpha 3, and beta 2 microglobulin. Okay, the peptide binding cleft between alpha 1 and alpha 2 in class 1 MHC molecule. Class 1 MHC molecule. Okay, and we have a this is called the membrane distal domains because it's distal to the membrane. This one, the membrane proximal domain. Okay, alpha 3, membrane proximal domain, proximal to the membrane, and we have a transmembrane segment. Okay, cell membrane of nucleated cell, and we have a cytoplasmic tail here like this. Okay, and class 2 molecule, in the class 2 molecule we have a alpha 1, beta 1, alpha 2, and a beta 2 microglobulin. Alpha 1 is there, beta 1 is there, alpha 2 is there, and beta 2 microglobulin is there. Okay, the peptide binding cleft is formed between alpha 1 and beta 1, formed between alpha 1 and beta 1. Okay, and a transmembrane segment is present like this one. Understood? So MHC1 has a smaller pocket for binding of antigenic peptide. MHC2 has a larger pocket for binding of antigenic peptide. Okay, it has a smaller pocket and it has a larger pocket. Understood? Here you can say small pocket is there. Here deeper cleft is there, so it is a larger pocket. Okay, so if I just I told you about the structure. Structure is very simple. You have to draw 4 circles like this. You can draw it like this. Okay, and you can draw a beta 2 microglobulin like this. For MHC1, I am drawing MHC1, alpha 1, alpha 2, alpha 3. Okay, just to move like this anticlockwise. Okay, and then draw a transmembrane domain. Okay, this is a transmembrane segment. Just draw the membrane here. This is the structure of MHC1. And if you want to draw the structure of MHC2, basically we have to do like this and a beta 2. Okay, so we have alpha 1, alpha 2, beta 1, beta 2, alpha 1, beta 1, alpha 2, beta 2, like this. Draw it like this. Okay, and here you draw 2 chains, here also and here also, and here's the membrane. Okay, this is the structure, and this is the antigenic binding cleft, and here this is the antigenic binding cleft. Understood? This is the whole structure. Okay, then MHC2 proteins are present on the surface of APCs. MHC2 is there. APCs express MHC1 as well as MHC2. MHC1 and MHC2 are both expressed by the antigen presenting cells. So MHC1 is the endogenous antigen or proteins that are present in the TC cells. MHC2 exogenous proteins expressed to the TH cells, not present, expressed to the TC cells. This MHC2 that present the exogenous proteins to the TH cells. So just understand MHC1. Okay, MHC1 is endo. Okay, endo, and TC means CD8. 1 into 8. I told you a rule of 8, right? 1 into 8, right? And 2 means exo. 2 means exo. This is in first means inside, just inner. This is outer. 2 means outside. Later one, outside one. And TH means CD4, right? This you have to keep in mind while you are reading immunology. MHC3, what about MHC3? MHC3 is also 10,000 kilobase per long, not involved in antigen presentation. Very important. It is not involved in antigen presentation. It has genes which are encoding various things. It is encoding these 8 things. This C2, C4, C3 convertase, factor B, properdin. This factor B properdin is basically regulatory molecules, and C2 and C4 are complement proteins. C3 convertase is an enzyme which will bring in the C3 and C3 convertase of different pathways of complements. We'll see and remember. C2, C4, C3 convertase, factor B, properdin are the complement proteins. HSP is heat shock proteins. This encode for heat shock proteins also, encode for TNF alpha beta, encode for steroid 21 hydroxylases. If you don't want to remember this, just leave it. Okay, understood? So this is the whole fact, and we'll discuss about the structure now and how the pathways are there. Okay, so a lot to discuss. Okay, no problem, we'll start. Yeah, so MSC3, did you understand about MSC3? MSC3 is 1000 kilobase per long, not involved in antigen presentation. Genes are encoding for C2, C4, C3 convertase, factor B, properdin. Okay, so these are the complement proteins which are encoding for heat shock protein, TNF alpha beta, and steroid 21 hydroxylases. Heat shock protein, TNF alpha beta. Remembered. Okay, chromosome 6p, this is the arrangement DP, DQ, DR of class 2 MSC. Then class 3, it is encoding for complement, TNF and heat shock proteins commonly, and class 1 means A, B and C. Okay, BCA. This is schematic representation of human MSC complex. MSC1, alpha 1 subunit has 90 amino acids, alpha 2 has 90 amino acids, alpha 3 also have 90. Beta 2 microglobulin is present. This is the whole chain. This is the transmembrane segment, cell membrane, cytoplasmic tail. Alpha 1, alpha 2, alpha 3 are called as extracellular domains because it's outside the cell and they're forming domain type of structures. Okay, so these are extracellular domains, right? Association of beta 2 microglobulin with alpha chain is needed for expression of MHC1 molecules on the cell surface. Okay, so beta 2 microglobulin will be associated with the alpha chain, and it is needed for the expression of MHC1. MHC1 is expressionless. MHC1 will not have any expression if this beta 2 microglobulin is absent. Okay, and what are Doddy cells? Doddy cells are DAUDI, Doddy cells, a type of human B cell tumor cell. Okay, B cell tumor cell, this is a type of tumor cells, but beta 2 microglobulin is not present. Such cells do not express MHC1 on the surface. They don't express MHC1 means MHC1 expresses endogenous pathogens. The endogenous antigens will not be expressed, so there will be infection and you won't know. So this is an example by which tumor cells are inducing the formation of the causing of another infections, the secondary infections, right? Okay, so this is the fact. And peptide binding cleft of MHC1, between alpha 1 and alpha 2 domains it is formed. Alpha 3 domain binds CD8 molecule present on T cells. Okay, alpha 3 domain is binding the CD8 molecule present on T cell. Okay, let's move on to the MHC2. Okay, let's move on to the MHC2. So MHC2 has alpha and beta subunit, and alpha again have alpha 1, alpha 2, beta has beta 1, beta 2. We'll discuss about that. So MHC2 has alpha and beta, alpha 1, beta 1 and alpha 2, beta 2, right, which have transmembrane segments and cytoplasmic tails. Is alpha and beta. Peptide binding cleft is formed between alpha 1 and beta 1. We just saw that you can draw it like this. Just a minute. This is the transmembrane tail, and here also we have a beta 2 that microglobulin is not there, like this. Okay, alpha 1, beta 1, alpha 2, beta 2, and peptide binding cleft is between alpha 1 and beta 1. Okay, so this is the basic structure of MHC2 molecule, right? And another thing is that 1000 kilobase per kilodalton that is there. Beta 2 domain interacts with CD4 molecules of TH cells, okay, during antigen presentation. So beta 2 domain is what which interacts with the CD4 molecule of TH cells. Very important. Peptide binding cleft of MHC1 is smaller, bind to 8 to 10 amino acid long proteins. MHC2 is bigger, which binds to 13 to 18 amino acid long proteins. and the antigen presentation by MHC1 is cytosolic pathway, and the antigen presentation of MHC2 is by endocytic pathway. Okay, so cytosolic pathway, and this is the endocytic pathway. So how can you remember? C comes first, after C E comes, so 1, after that 2 comes, right? You can remember like this, just a mnemonic. Okay, so let me tell you the pathway of antigen presentation by a cytosolic, and then we'll go to the endocytic pathway. So first come to cytosolic pathway. So cytosolic pathway, what happens is that we have an endogenous protein. Okay, this is a pathogenic endogenous protein, and this endogenous protein is processed by fed into the proteasome. Okay, this is microbial protein fed into the proteasome. This proteasome eats it. This proteasome converts it into peptides. This forms small peptides of this protein. After the small peptides, it is transported into the endoplasmic reticulum. In the endoplasmic reticulum, it goes via the tap. Remember in this picture also, in the chicken wala picture also, we saw that tap. Okay, this tap is basically transporter associated with processing. Okay, transporter associated with processing. It transports the peptide fragments inside the endoplasmic reticulum. Okay, and here the endoplasmic reticulum, there is the MHC molecule present, MHC1 present. Okay, and MHC1 peptide complex is formed. Here's the MHC1, and here the MHC1 peptide complex is formed. This MHC1 peptide complex is packaged inside the goes to the Golgi apparatus, where it gets sorted and all, and after that it is exocytosed. Exocytosis means MHC class 1 molecule will be there with this peptide fragment. The small fragment is the peptide fragment, right? This one, and so this is formed, and it is presented to the CD8 T cells. Easy, right? Yeah. Okay, this is the thing. This is the same thing, just a comical representation of the same thing. Okay, and after that we'll see the endocytic pathway. That was the what pathway? Your cytosolic pathway. Cytosol is the endocytic pathway. Let's move on to the endocytic pathway. This is a little bit complex. Just don't worry. We'll deal with it easily. Okay. Okay. These things are there now. Yeah. So in endocytic pathway, some things are occurring simultaneously. Just try to understand here. In the endoplasmic reticulum, MHC2 binds to the invariant chain protein. Okay, basically when an exogenous antigen, we ingest an exogenous antigen. Okay, and bacteria comes and infects, and exogenous antigen is present, or a bacteria is here, or whatever. Then it fuses lysosome. That phagosome formation all will occur. Then endolysosome is formed. This endolysosome. This endolysosome. Okay, in the endolysosome, it goes to the endoplasmic reticulum, where MHC2 binds with invariant chain protein. A protein is there called invariant chain protein, form the invariant chain MHC2 complex. Forms the invariant chain MHC2 complex, which is secreted into the sac and forms the endosome. Okay, endosome. This invariant chain MHC2 complex are packed, are secreted into the sac and form the endosome. Understood? This part done. In this endosome, an enzyme is present where the invariant chain is broken down into 3 fragments, where the largest fragment is called as the clip. Okay, so what is happening? Your MHC2 is binding with invariant chain. Your invariant chain MHC2 complex are now secreted into the sac. This sac fuses the endosome, or forms the endosome. Okay, where the enzyme is present. What enzyme? The enzyme which will break down this invariant chain. This invariant chain is broken down into 3 fragments. Of these 3 fragments, the largest fragment is called as the clip. Okay, this is the largest fragment, is called as the clip. This clip, okay, just understand up to this. Then after that, we told that exogenous antigen has entered. This exogenous antigen has will form the phagosome. Okay, this form the phagosome, where the peptide fragments are present. Okay, the fragments are broken down. This clip and the here clip is there and MHC2 is there, right? Because invariant chain and MHC is binding now. So largest fragment is the clip. So clip has bound to the MHC2. Now this both will fuse. This both this complex and this peptide fragment will fuse, and this clip will be replaced by this peptide fragment and MSC2. Now this will be getting presented. Now this will come and transported by cell membrane, exocytose, and will be presented. Okay. So here a little bit complex pathway, this endocytic pathway is that. Okay, and but it's easy to understand, not that difficult. Okay, let me repeat the concepts. Okay, so here we'll understand both endocytic and cytosolic pathway, and I will repeat it for you. Okay, it will take time, this immunology will take time, but I am trying to cover all the concepts which are there for you to understand. Okay, so basically we are in the cytosolic pathway. First, how we will draw the cytosolic pathway. Okay, so we have an endogenous antigen. Let's say this is the endogenous antigen. This endogenous antigen is now fed into what? Fed into a proteasome. Let's say this is the proteasome. Proteasome will convert it into small fragments. These are the small fragments. Okay, we have the endoplasmic reticulum here. Endoplasmic reticulum will have a protein. What protein was there? TAP. What is TAP? TAP is the transporter associated with processing, right? This TAP will transport it into endoplasmic reticulum. In the endoplasmic reticulum, what other thing is there? Your MSC2 molecule is there. Okay, let's say here we have the MSC2. The MSC2 will bind to this protein fragment. Let's say this is the protein fragment. It will bind to the protein fragment like this. Okay, this is bound, and then it will be sent to the Golgi apparatus. So here we'll draw the Golgi apparatus like this. Just I'm drawing it very roughly. In exams you may color the diagrams and all. Okay, here it is packaged into this part. Okay, it's packaged, and here it is exocytosed, and this will be presented out like this, the peptide fragment and this all, and here there will be CD8 T cell. Okay, be recognized by this TCR. Understood? This is the whole cytosolic pathway. Understood? And after cytosolic pathway, let's move on to the next pathway, that is your endocytic pathway. We discussed the endocytic pathway, then endocytic pathway. So talking about the endocytic pathway, what was there occurring in this pathway is that first thing in the endoplasmic. Let me draw a cell and show you. Okay, in there are the changes in the endoplasmic reticulum. Let's say this is the endoplasmic reticulum. In the endoplasmic reticulum, the MHC2 will bind to the invariant chain. Let's say this MHC2 was there. Let me draw the MHC2 like this. MHC2 plus invariant chain protein. They're bound. Okay, then these are secreted into the sac. Then this go to the Golgi apparatus here. Golgi apparatus is there. Okay, secreted into the sac. In this sac contains MHC2, and let me draw the invariant chain in red. Okay, so there is a largest fragment of invariant chain which is called as the clip. So invariant chain has a largest fragment which is called as the clip. So this clip and MSC, this is the MSC2, right? So this clip and MSC2 are bound here. This is your how the clip is formed? This clip is formed because this invariant chain is broken down. Okay, broken down. So this is the thing. Okay, and another thing is that when the exogenous antigen has entered, this is the, let's say this is the exogenous antigen which is entering. When this entered, it fuses with the lysosome. This lysosome, you can draw all the parts here. Okay, and here we draw finally the peptide fragments. This is called the phagosome. Okay, this is a phagosome, this is the endosome. Here what happens, this one both will fuse, and this let me draw the endogen this proteins in purple because color coding, let me maintain it. And this clip is replaced by MHC2. Sorry, this MHC2, this clip is replaced by this antigenic fragment, that is a peptide fragment, and this whole part will be presented. Okay, so this is the endocytic pathway, means inside the cell is all are occurring, and this is the cytosolic pathway, in the cytosol all this occurring. Okay, both pathways I made you understand, right? Both pathways are done. Okay, let me delete the page. Yeah, after that some other things are there regarding MHC, and then we'll continue. MHC proteins are polymorphic proteins, that is MHC1, HLA A, B and C are there. HLA A has 450 or 500 alleles, 500 you remember for more here. HLA B has 800 and HLA C has 250, just half it, 500, 250. HLA A and HLA C are just half, HLA C is just half of that. HLA B has 800 alleles, okay. And MHC2, HLA DP, DQ and DR is polymorphic due to multiple alleles, polymorphic due to multiple alleles, okay. So MHC1 is there, MHC2 is there, MHC proteins are polymorphic proteins, and these are the things, okay. And MHC proteins have a codominant expression, important codominant expression, codominance is there, okay. That is 2 alleles, like MHC1 chromosome P, HLA A. Let's say this is the MHC1 on chromosome P, HLA A. So HLA A1 is there paternal, which is HLA A8 maybe, okay, any one of them. A8, A9, any one of them. So one is paternal, one from maternal. So they have codominant expressions. Okay. So just remember, whenever there is a question on MHC, you write that for the MHC proteins are polymorphic. Polymorphic means you can write HLA A and what are the number of alleles. HLA A has 500 alleles, HLA B has 800, HLA C has 250, right? Then you write that MHC proteins are codominant expression. MHC1 paternal, maternal, you could write. Professional APCs, I told you that dendritic cells, macrophage B cells, dendritic cells are the best, okay. Why they are the best? Because they constitutively express increased level of MHC2 molecules and B7 molecule. B7 is a co-stimulatory molecule, we'll discuss about that. Macrophage need to be activated by phagocytosis microbes, so they are just the second best, okay, you can say. And B cells constitutively express MHC2 molecules, but they need to be activated by internalization of antigen. So first, for activation of B cells, there must be an internalization of antigen before they start to express B7. So they are just, you can say, least good. Some non-professional APCs are there, very important to remember. This part is also important, this slide is also important. Cells is normally don't express MHC2 adequately are called as non-professional APCs. Okay, normally don't express MHC2 adequately, but can be induced to do so under chronic conditions like chronic inflammation. Okay, they are working as antigen presenting cells all the time, but they inadequately express MHC2, but they can be induced to do, they can be made to induce to express in cases like chronic inflammation. Okay, so we have fibroblast, vascular endothelial cells, glial cells, thymic epithelial cells and pancreatic beta cells. Okay, what are the things? You have fibroblast, vascular endothelial cells, glial cells in brain, thymic epithelial cells and pancreatic beta cells. So these are the non-professional APCs. With that, we complete our MHC molecules class, and let's move on to the next topic now. Let's discuss about pattern recognition receptors now. So these are abbreviated as PRRs, present on immune cells like macrophages, and patterns which are present on microbes are referred to as pathogen associated molecular patterns, PAMs, okay, and or microbes associated molecular patterns, MAMs. So PAMs and MAMs are present on microbes, right? They are present on the surface of microbes. And if microbes are dead, let's say dead organisms are there, dead microbes are there, so we have damage associated molecular patterns. So this PAMs, MAMs and DAMs all are of same category basically, but this differ in the terminologies. Pathogen, these are microbes are pathogens only, so these are used synonymously, but if the microbes are dead, then DAMs, okay. And best examples of PRRs, pattern recognition receptors, is TLR, toll-like receptors. You must have heard this term previously, or if you haven't, the toll-like receptor. So toll receptors in Drosophila were discovered in Drosophila, are the main receptors for innate immune response in Drosophila. Okay, these were first discovered in the Drosophila, right? And are similarly present in us also. Similar receptors are present. About 13 TLRs are present. which we have extracellular TLRs and intracellular TLRs. Extracellular means we have 1, 2 and 6, TLR 1, 2 and 6, which recognize the bacterial lipopeptides. Okay, the bacterial lipopeptides. So number 1 movie Special 26 was a movie. You can remember like this: number 1 movie Special 26, right? 1, 2 and 6. Okay, then we have intracellular TLRs. So TLR 2, 4 and 5, these are recognizing bacterial peptidoglycan. 2 is recognizing bacterial peptidoglycan. Okay, 4 gram LPS, gram negative gram negative. You can just remember like this: RODS, RODS, gram negative, GRAM, 4 letters, so 4. Bacterial peptidoglycan, 2 letters, B and P, so 2. Okay, you can remember like that. Or 5 is the flagella bacteria. 5 you can write like this, right? 5 you can write like this. So this is in the shape of a flagella. So 5 is for recognizing the flagella bacteria. 2 is recognizing bacterial peptidoglycan. 4 is recognizing LPS of gram negative, and 5 is flagella bacteria. Okay, let's discuss the intracellular TLRs. So intracellular TLRs, we can see TLR 3, TLR 7, 8 and TLR 9. So 3 is recognizing dsRNA. So 3D, you can remember like this: 3D, remember 3D. 7 and 8 are recognizing the ssRNA of viruses. So viruses which have a single stranded RNA, these are recognized by TLR 7 and 8. TLR 7 and 8. Okay, dsRNA are recognized by TLR 3. And TLR 9 is recognizing bacterial DNA. Okay, TLR 9 is recognizing bacterial DNA. B you write like this, so you can just reverse it from 9. So 9 bacterial DNA, dsDNA of herpes simplex virus also. Okay, and binding of TLR to MAMS, if a TLR binds, there's the activation of transcription factors, which will stimulate the expression of genes encoding transcription factors. Okay, increase the expression of genes encoding transcription factors. Example is the nuclear factor K beta and interferon regulatory factors IRFs. Okay, so cytokines and interferons, these are the transcription the coding for cytokines and interferons. Okay, so basically this TLRs, binding of TLRs to MAMS, are causing activation of transcription factors, which are stimulating the genes producing the transcription factors. Example of gene are nuclear factor K beta gene, interferon regulatory factor IRFs. In the nuclear factor K beta produces cytokines, and interferon regulating factor IRF produces interferons. Next we have NOD-like receptors. So toll-like receptors is done. Let me quickly go through extracellular 1, 2 and 6, 2, 4 and 5 also. Okay, 2, 4 and 5, 1 to 6 and 2, 4, 5. 2, 4, 5, I told you the tricks. 1, 2, 3 recognizes bacterial lipopeptides. Okay, 2 means bacterial peptidoglycan, 2 letters. 4 means LPS of gram negative, bacilli rods. Okay, and 5 is like a flagella, so 5 is the flagella. Intracellular TLRs, we have 3, 7, 8, 9. 3 is 3D, dsRNA of virus. dsRNA important, ds not DNA, RNA. Okay, RNA. And 7 and 8 with the ssRNA of virus. Okay, ssRNA 7 and 8. Okay, and TLR 9 bacterial DNA. TLR 9 is for bacterial DNA. B you write like this, and DNA like this. And herpes simplex virus. Okay, binding will activate the transcription factors, stimulate the expression of genes encoding this transcription factors, like nuclear factor K beta and interferon regulating factor. Nuclear factor K beta codes for cytokines, IRFs code for interferons. Then we have NOD-like receptors. NOD-like receptors mean part of peptidoglycan bacteria recognized by NOD-like receptors, which is present in the cytoplasm of human cells. Which human cells? MED. Remember MED, macrophages, epithelial cells and dendritic cells. Epithelial cells and dendritic cells. Macrophage, epithelial cell, dendritic cell, MED cells. Okay. So peptidoglycan bacteria is recognized by NOD-like receptors, which is important in innate immune response to intracellular bacteria like Listeria monocytogenes. Okay, so NOD-like receptors are helpful for innate immunity against Listeria monocytogenes. Okay, understood? NOD-like receptors is done. Then last one we'll discuss about Rig1 helicase receptor. Rig1 helicase receptor recognizes nucleic acid of viruses and cytoplasm of infected cells. It recognizes nucleic acid of viruses. So first TLRs, toll-like receptors, are recognizing various functions. Bacterial lipopeptides basically 1 to 6. 2 was recognizing bacterial peptidoglycan. 4 LPS of gram negative. 5 flagella. Intracellular 3 was dsRNA of viruses. 7, 8 ssRNA of viruses, and 9 is bacterial DNA. Okay, And node-like receptors we have recognizing innate important in innate immune response in intracellular bacteria against intracellular bacteria Listeria monocytogenes, okay, and present on mate cells. And RIG1 helicase receptor recognize nucleic acid of viruses in cytoplasm of infected cells, like in members of rhabdoviridae, orthomyxoviridae, and paramyxoviridae. Okay, these are genome ssRNA. So in members of this rhabdoviridae, orthomyxoviridae, and paramyxoviridae, okay, synthesize and manufacture dsRNA during replication, which is recognized by RIG1 helicase receptors. So dsRNA, they are recognizing dsRNA. Okay, basically these are ssRNA viruses. Rhabdoviridae, orthomyxoviridae, and paramyxoviridae are ssRNA viruses, but these are recognizing, okay, dsRNA. They are recognizing dsRNA, which is recognized by RIG1 helicase receptors. Okay. When microbes are phagocytosed, they end up in phagolysosomes. During destruction in phagolysosomes, their codes are stripped off, internal molecules are revealed and recognized by TLRs and phagolysosomes. TLR7 recognizing ssRNA viruses, we know this. TLR7 we told about this, yes, ssRNA viruses, okay. And TLR9 dsRNA bacteria. Understood? Bacterial chromosome, okay, dsRNA basically. We told about this, right? Bacterial DNA, yes, this one. Okay, B for bacteria, B for TLR9. Okay, point inverse through phagolysosome, you can just leave it. PRRs have two important possibilities: recognize general characteristics of microbes. They are basically recognizing general characteristics, okay, not specific ones, like LPS and ssRNAs. Recognize structural molecules of microbes which can be easily affected by microbes using mutation. Okay, they recognize such molecules which the microbe cannot easily change, by mutations. Like TLR4 recognizes LPS. LPS can't be easily changed by the bacteria by mutations. Okay, so this completes the pattern recognition receptors. So we learned about what are pattern recognition receptors, what are the binding, what TLRs are there. 13 TLRs are there: intracellular, extracellular, 6, 30, and 3, 7, 9. And remember the important ones. Binding, what are the effects on binding? Node-like receptors, what are they? Okay, peptidoglycan bacteria, recognize them. And RIG1 helicase, about nucleic acid of viruses and all. dsRNA, recognize dsRNA. And then two important functions we studied about general characteristics and structural molecules which can't be easily altered by mutations. So that completes the PRR. We'll move to the next part now. Now let's move on to the antibodies. So what are antibodies? Antibodies are immunological molecules which bind with its corresponding or cognate antigen and induce the production and participation of immune response. Okay, so antibodies are binding with the corresponding antigen, and induce their production, also induce the production of this antigen, and also participating in the immune response, right? So if these antibodies are acid, this type of if the plasma proteins basically are acid, this type of curve is there when the highest peak is obtained by albumin, because albumin is the most abundant, right? And then alpha, beta, and gamma globulin. So antibodies are gamma globulins, but all gamma globulins are not antibodies. All gamma globulins are not antibodies, but all antibodies are gamma globulins, right? The statement is important, right? Then antibodies are also known as immunoglobulins, and chemical name is glycoproteins. They are glycoproteins. Glycoproteins means prefix is always less in amount, okay, and suffix is always more in amount. So glyco means less of carbohydrate and more of protein. So very less carbohydrates is also present. Majority of function of an antibody are by virtue of its protein part. Okay, this protein part, which is more in number, obviously will have majority of the functions, right? Very obvious. Y-shaped molecule, 4 protein chains are there, 2 heavy chains, identical 2 heavy chains, 2 identical light chains joined together by disulfide bonds. You know all this from your previous classes, I hope. Okay, structure of a monomeric antibody. Okay, there may be many mechanisms like dimeric antibody, pentameric antibody. We'll see about them, but we are discussing the structure of a monomeric antibody. So this is the whole structure. Here you can see this yellow shaded part. Okay, this yellow shaded part is the heavy chain. Okay, this is the one heavy chain, and this is one heavy chain, this is one heavy chain. Okay, and about light chains is blue shaded part. This is one light chain, this is another light chain, right? And this heavy chains and light chains are also have different parts, like this VH means variable region of heavy chain. This is the heavy chain, right? And this is the variable region of heavy chain, and this whole part is the constant region. Constant region is also divided into CH1, CH2 and CH3. Three parts: CH1, CH2, CH3. And again this same thing in this side: VH1, CH1 and CH2, CH3, right? And in the heavy chain we have VL is the variable region of light chain, and Cl is the constant region of light chain. Cl is the constant region of light chain. Here between the VH and VL part we have the antigen binding cleft, or the paratope. You know about the paratope, right? Epitope was present on the antigen, like this. We have a diagram. Here is the epitope, and paratope was present on the antibody. This is the NH2 side. Here the NH2 molecule is there, and here is the COOH side. Done. This is the antigen. You can see this whole thing. Okay, done. Okay, apart from interchain SS bonds, intrachain SS bonds are also present inside the L and H chain. Okay, this is the interchain. Okay, this is the interchain, and this is the intrachain, and interchain bonds are also present, like this. This type of bonds are also present, which cause the formation of domain. Domain formation is done by this interchain hydrogen bonds. Okay, two types of light chains are there: kappa and lambda. Okay, this light chain also, this whole part, are of two types. This are of kappa type, or this may be of lambda type. Lambda type. I'm writing gamma, kappa and lambda. Okay, a single antibody molecule cannot have both kappa and lambda. It may be of one type, either kappa or lambda. Okay, 2/3 of antibodies circulating in blood are of kappa chains. Kappa is more, you can say, abundant, right? And 1/3 of antibodies have this lambda chain. So kappa is to lambda ratio is asked in exams, 2:1. Genes for kappa chain are present on chromosome number 2. K for kappa, K for and 2. Okay, genes for lambda are present on chromosome number 22. Okay, you can remember this. So 2 and 22, very easy to remember. Genes for heavy chain are present on chromosome number 14. Okay, because 4 plus 1 is 5. 5 means heavy. Okay, you can remember like this. Understood? So 14, chromosome number 14 has a gene for heavy chain. Gene for light chain, kappa chain, is present on chromosome number 2. Gene for lambda is 22. 5 classes of antibodies are there: IgG, IgA, IgM, IgD and IgE. Okay, 5 classes of antibodies are there. This is the order of serum concentration, GAMDE, GAMDE. You can remember this statement. IgM and IgD, IgM and E, right? Me have longer heavy chains. Okay, longer heavy chains means there are no hinge regions. Hinge regions, you know, this part is known as a hinge region. Here the antibody can turn over itself and be rebounded back over itself. This is the hinge region. And here in the IgM and IgE, hinge regions are absent, but they have longer heavy chains. That is 4 CH regions are present: CH1, CH2, CH3, CH4. Normally 3 were present. Okay, right, this CH1, CH2 and CH3, but here there are 4 present, right? CH1, CH2, CH3 and CH4. Antigen binds to the VH-VL region and not VH alone or VL alone. So antigen is basically binding to the VH-VL region. VH-VL region means you are understanding, right? Here's the VH, here's the VL. So antigen is binding beneath here. Okay, not in the part. So number of VH-VL regions in an antibody molecule is called as valency. Okay, valency is also defined as the number of antigen an antibody can bind to, but the most accepted definition microbiologically or immunologically is the number of VH-VL regions in an antibody molecule, right? Then monomeric antibody means 2 VH-VL regions are there. Valency is 2 because 2 antigens can be bound to it. Paratope, what is paratope? VH-VL regions are forming the paratope, combines with the epitope. We know this. Within the variable regions are some hypervariable regions are present, or hotspot, or idiotypes. Hypervariable region, hotspots or idiotypes are present. Here the exact precise bond is formed between antigen and antibody. Right, you can see this in the light chain. In this light chain also, in this light chain and in the heavy chain and light chain. Okay, some hyper variable regions are there. In this diagram is basically, just a minute. Okay, this diagram is basically from your Warren Levinson. So in this, what is the problem is that light chains are this one are the light chains and this is the heavy chain. Of course it isn't just a representation, so it doesn't matter which one is light or which one is heavy. Okay, basically this heavy chains are CH1, CH2, CH3. This is the main thing which should be done. You may draw it like that, or you may draw it inside also, no problem in that. Okay, so light chain hyper variable regions are there. You can see hyper variable regions are these ones. Okay, so and hyper variable regions of light chain and hyper variable regions of heavy chain. Okay, there are these sites are known as your hotspots or hyper variable regions or idiotypes, right? Here a precise bond is only formed between these regions. What are the precise bonds? HIV, you can remember, hydrophobic interaction, hydrogen bond, ionic bond, and van der Waals forces, right? Antigen antibody reaction is firm but reversible. They are reversible, not irreversible. An antigen is bound to the antibody and irreversibly, but it is firm. Okay, it is strong. Action of proteolytic enzymes. This is an important part because the previous things you have read in your previous classes, but this part may be new to you, or if you have read in biochemistry, then well and good. Let me talk about this, then we'll talk about different allotypic determinants and other antibodies in detail. Okay, let's talk about this first. So second thing, so if I talk about proteolytic enzymes, we have papain and pepsin. Two enzymes are there. What is the function of papain? Papain means A is coming, right? So above, you can remember like this. So this enzyme will cut the antibody above the heavy chain disulfide bond. Means we have three disulfide bonds. Let's assume for simplicity 1, 2 and 3. Here three bonds are assumed, and this is the heavy chain disulfide bond, and papain is cutting here. So basically three fragments are generated, 1, 2 and 3. You can see this fragment is called as FAB. This is also called as FAB because if you put some antigens, they will bind to this parts only. So it was named FAB by this action of papain only, and one FC fragment. So two FAB fragments. Action of papain is that it will generate two FAB fragments and one FC fragment. This is the question. Okay, it's the answer of the question. Each FAB has a valency of 1. Let's discuss the action of pepsin next. Okay, now discussing about pepsin, action of pepsin on antibody. Here we have the antibody like this. This is the antibody, and here are the disulfide bonds present between them. So pepsin is basically cutting it from downwards because papain was cutting it from up the disulfide bond, and here it will cut like this. Okay, if it cut like this below the interchain disulfide bond, then one segment is this one, which is known as FAB2. It is bound to two antigens which we are giving here, and two fragments are there, so further broken down into amino acids and are known as FC des. Okay, so action of pepsin on antibody generates FAB2 whole 2. Okay, not two FABs, FAB whole 2 plus one FC des fragment. Okay, this is the question which you have to remember. Talking about isospecificity, known as class or a isotype, have nothing to do with specificity of antigen antibody reaction. Isospecificity is determined by CH reasons. Basically we have 5 types of isospecificity. If this is the order of serum concentration, gamma day, you know. So 5 types means 5 classes or isotypes or isospecificities. So CH reasons is of gamma type, G means gamma type, A means alpha type, M means mu, D means delta, and epsilon means IgE means epsilon. Okay, these are the types of CH reasons. So these are specific for this. Okay, like IgA has 2 subclasses, IgA1 and IgA2. This is the order of serum concentration. IgG has 4 subclasses, IgG1, G2, G3, G4 in this order of serum concentrations, right? And class is determined by the CH reasons. So this is the isospecificity that is determined by CH reason, and these are the things which you have to remember. So these are the isotypic determinants, mouse IG1 and mouse IgM. These are entirely different entities, but here just note that these all parts are different, these all parts are different, this is different, this is different, which means that you have the constant region of the heavy chain which is different in this both, and this light chain this variable region of heavy chain as well as variable and light region of light chain are the same, right? Next we talk about allotypic determinants. Allotypic determinants they are also known as allo types. Okay, we discussed about isotypic determinants, and now we have to discuss allotypic determinants or allo type. Refer to the variations in certain amino acids in certain regions of CH part of antibody class amongst different members of a species. Now, okay, like we have to see mouse IG1 strain A and mouse IG1 strain B, means two different members of a species are compared here. In allotypic determinant, allotypic itself means that we are comparing two members of same species, right? Understood? So this is the thing. And mouse IG1 strain A and mouse IG1 strain B, here you can see the CH region are almost the same. This one, this circle and triangle same, but this rectangular part is different from this. Okay, you can see here is that these are called the allotypic determinants. So these are compared in the same class of an antibody is being analyzed, right? So these are the allotypic determinants. Understood? So these are seen in kappa light chain KM system. 2 allotypes are there. GM system 25 allotypes and IgA light chain AM system. Okay. So KM system examples you can write KM system 2, GM system 25 allotypes, and AM system also. Okay. AM system numbers are not specific, not required for you. In the past, knowledge of allotypic determinants was helpful to solve the cases of paternity disputes. Understood? Next type of idiotypic determinants, which are BH-VL regions, okay, which are also known as paratopes. Paratopes are like this paratopes are also known as idiotypic determinants. They are also determining which antigen is being bound to this BH-VL regions. We know that antigens get bound to this BH-VL regions, right? So you can see from here idiotypic determinants. Just a minute, yeah. So mouse IG1 and mouse IG1 same antigen A against antigen A and against antigen B. Here are the idiotypes against antigen A. You can see this type of allotype is there, and this arrow type and this circular type. These are the allotypic idiotypes of antigen A. And of antigen B, what are the idiotypes? This rectangular one is there, another this type of there, and this one. Okay, these are of antigen, these are against antigen B. Okay, idiotypic determinants. Next, there are 3 rules for antibodies. So we are discussing the rules for antibodies. What are the 3 rules? And then we'll go to the individual antibodies, right? So what are the rules? First thing, a single B cell is monospecific. Means that if a B cell is there, just assume there's a B cell here. Assume there is a B cell, as the BCRs are there, this get differentiated into a plasma cell. This is a plasma cell having more Golgi apparatus, more secretory functions, well developed Golgi apparatus, and a peripheral nucleus. You know about this is a plasma cell. This is a plasma cell. Production rate is 2000 antibodies per second. We discussed about that. It will produce antibodies. The antibodies it will produce are exactly similar to that of BCR. Exactly similar. And bind to a single type of epitope. This is known as your monospecificity. So a B cell is monospecific. Understood? B cell is monospecific. This is the thing. And let's move on to the next part. So next thing is that if one plasma cell secretes antibody of one class only at a time. Okay, means if this is a B cell, it will the plasma cell will secrete only one class of antibody, like IgM. If it is secreting IgM, then it will secrete IgM only. Okay, so if this is the B cell, then this is the BCR. If the plasma cell is there, only IgM will be produced, or IgG will be produced alone, not IgM plus IgG. This is not possible. Or IgA, not possible. Okay, so only one class of antibody is produced by one plasma cell, and a class can be switched by a plasma cell. Any other class is not made, but if class can be switched, like if IgM was being produced, it can after some time switch to IgG, because you know IgM was initial antibody and IgG comes wherever there is a chronic reaction. by time. Okay, so IgM synthesized by that plasma cell stops, but the same plasma cell now can produce IgG. This is called as class switching. So class switching, you know this. Rule 2 is still followed, only one class is being produced, that is IgG, not a combination. Okay, you can't combine this, but can be switched. Okay, during class switching, it is the CH region which changes and not the BHBL region. So CH region is basically the determining the class, because we know that CH region are basically determining the class, right? This is the B cell and this is the BCR, and the antigen is present over here. If it gets differentiated into a plasma cell, then IgM antibody is being produced and the same antigen is bound, right? In the class switching, this constant part, the CH part is being changed now. The CH part is being changed now. There is no difference in the VH and VL region. This constant part is basically determining which class of antibody is basically produced. Okay. Let's discuss the concepts of the biological functions of antibodies. What are the biological functions of antibodies? And biological function depend mainly on the isotype, class or isospecificity. Depend mainly on class and isospecificity. So biological functions, we will discuss each antibody in detail. So let's start with the IgM antibody. IgM, M stands for millionaire molecule, because molecular weight is 10 to the power 6 Dalton. Okay, molecular weights you don't have to remember much, but if it comes in the name, then you should have knowledge. Pentameric structure is there. This type of pentamer is present, and the GA chain connects this pentamer. Valency is 2, because 2 antigens can theoretically bind to here, right? 1 and 2, right? So theoretical valency is 10, because 5 into 2, right? 5 into 2 is 10. Then actual valency is 5, because there is steric hindrance. Just assume here is one antigen, here is one antigen. There is a steric hindrance between these two. Okay, there is a steric hindrance present between these two, right? So steric hindrance. So the theoretical valency is basically 5 due to steric hindrance. Most of the IgM, 80%, is present in intravascular component, because it is a very big thing. Okay, so if it gets inside the circulation, it can't leak out very easily. Okay, so most of them are present in the intravascular component, 80%, right? It is difficult to cross and go out due to its size. IgM is the antibody of acute inflammation or recent inflammation. I told you it is the initial antibody, first antibody produced in any infection. The first antibody produced in any infection is IgM. The first antibody produced in any infection is IgM. Very important statement. Okay, only class of antibody synthesized before birth. It starts about 20 weeks of gestation. The synthesis starts about 20 weeks of gestation. Okay, 20 weeks of gestation. So IgM is the antibody of recent or acute infection. First antibody produced in any infection. Only class synthesized before birth. Okay, class produced 20 weeks of gestation. Produced at 20 weeks of gestation. IgM is the most potent activator of classical pathway of complement system. Most potent activator of classical pathway of complement system. Right. We'll discuss about the complement system in a little bit detail later after this topic. Also known as agglutinating antibody, because pentameric structure is there, right? So if it bounds to the RBCs, it will clump the RBCs. Agglutination reaction, you know, and this agglutination reaction is basically caused by this IgM. So it is known as agglutinating antibody. Also known as lytic antibody, because after agglutination there will be lysis, right? Following it, clump formation is being done. IgM is present as BCR, SIG or MIG on the surface of B cells. So BCR, SIG and MIG, here IgM antibody is present sometimes. Okay, SIG and MIG, you know, soluble antigen and all. Okay, so this were the things about IgM. IgM, first thing, IgM means millionaire molecule, 10 to the power 6 Dalton. Then you have to discuss its structure, pentameric rosette, JSF structure. Just close your eyes and listen to what I am saying. And the valency is 10, actual valency. Then theoretical valency is basically 5. Most of the antibodies, IgM is present in the intravascular component. Very less is present extravascularly, 20%. Okay, and the major thing is that it is the antibody of acute or initial recent infection. It is the first antibody to be produced. The first class of antibody synthesized before birth at 20 weeks of gestation. And IgM is the most potent activator of classical pathway of complement, known as agglutinating or lytic antibody, because it can clump and it's present on the surface of B cells as BCR, SIG or MIG. Okay, done. Let's talk about the IgG antibody. IgG, G means gun by. Gun by means antibody of past or chronic inflammation or secondary immune response. It's antibody of past infection, antibody of chronic infection, or antibody of secondary immune response. Very important. Okay, It's the longest serum T half, longest half life, 23 days. So it basically regresses or dies after 46 to 50 days, right? So it's the longest serum T half, 23 days. It's the maximum serum concentration, because longest T half means obviously there will be maximum concentration in the serum, right? It is a monomer. Valency is 2, equally distributed between intravascular and extravascular. Valency 2 means it can easily go out as well as in the intravascular and extravascular components across them. Only class of antibody that can cross placenta, because valency is 2, they are smaller in size. Okay, they can cross placenta. IgG2 is a bit larger, so it cannot cross placenta. IgG2 cannot cross placenta, right? And IgG is the second most potent activator of classical pathway of complement. Second most potent activator of classical pathway of complement, right? So we have basically 4 subclasses: IgG1, G2, G3 and G4, order of serum concentration. IgG2 subclass, the maximum allotypic variation is present, and IgG2 I told you also that it cannot cross placenta. IgG2 cannot cross placenta, I told you. And IgG is a good option in opsonizing antibody. IgG1 and G3 are much better opsonins as compared to IgG2 and G4. Okay, so odd numbers are better opsonins, just remember like that. IgG is a precipitating antibody. Precipitation reaction and agglutination reaction, we told about that, right? So basically this antigen, this IgG is basically precipitating at the bottom. In a newborn, which class of antibody is found most commonly? It's a IgG, because it can cross placenta. Most common antibody synthesized by the baby is IgM. Okay, synthesis wise, it is the IgM which is the most common, and in newborn the most common antibodies is found is IgG. Just remember these two questions distinctly, don't get confused. So this finishes our IgG. So basically close your eyes and let me repeat for you. IgG was gone by means it's the antibody of past or chronic inflammation. Longest serum T half, that is of 23 days. Maximum serum concentration, it is a monomer, it's the smallest antibody, it can cross the placenta, except IgG2 which cannot cross the placenta. It is equally distributed due to its smaller size, and second most potent activator of classical pathway. Okay, IgG4 pathway, IgG4 subclass activates the alternate pathway. Just remember this, alternate pathway is activated by IgG4 and classical pathway is not activated by it. It has 4 subclasses: IgG1, G2, G3, G4. Serum concentration order is this. IgG2 has the maximum allotypic variation. IgG is a good opsonin or opsonizing antibody. G1, G2 have much better opsonins as compared to G2 and G4. IgG is the precipitating antibody. Okay, and in newborn which is present IgG, and most common synthesized by the baby is IgM, right? Let's talk about IgA antibody, local antibody, mucosal antibody or secretory antibody. Local, mucosal or secretory. Local, mucosal and secretory antibody. Both three are the same names. Local antibody because it is acting locally in the present the local secretions of the body. Mucosal because mucosal surfaces are secreting it, and secretory because it is present in most of the body secretions, right? Like colostrum. It is present in large amounts on mucosal surfaces. Most common class of antibody found in the breast milk and colostrum is the IgA. You must have been knowing this from your pre-NEET times. Okay, and IgA activates the alternate pathway. Just A for alternate, right? Alternate pathway is activated by IgA. IgG was activating alternate pathway, but IgG4 was activating the alternate pathway. Okay, IgG was activating the classical pathway. A is activating the alternate pathway. Two subclasses are there, IgA1 and A2. Okay, this is the order of serum concentration. A1 is greater than A2. IgA exists in two forms. Two forms means serum IgA monomer, valency is 2, and secretory IgA dimer, valency is 4. Okay, serum IgA valency is 2 and secretory IgA valency is 4. Understood? So it is present on mucosal surfaces. So two forms, serum and secretory. Structure of secretory IgA, this is the structure of secretory IgA. Two monomers are there and linked by the J chain. So J chain is the okay, this is two monomers and J chain are produced by the plasma cells only. Okay, and there is a secretory component or secretory piece like this linking this both antibodies, like it's a dimeric structure, right? So this is the secretory IgA, valency is 4. You can see here the 1, 2, 3 and 4, valency is 4, right? So this is the thing. So 4 valency means there must be a J chain that must be linking, and a secretory component just produced by the mucosal epithelial. So how it is produced, just see here. Okay, Here's the J chain. This is the basic molecular structure. You don't have to remember the molecular structure. You can draw this type of structure once it comes in the exam. And yeah, so this is the J chain, and how it is produced. Just let me tell you. Secretion of secretory IgA. So we have the plasma chain here. Plasma chain will synthesize this J chain and dimeric IgA, right? Dimeric IgA, this type. We told this, right? Here, two monomers and the J chains are produced by the plasma cell. So dimeric IgA and J chain is produced by the plasma cell. Then we have a poly Ig receptor present on the mucosal cell surface. This is the mucosal cell. This is the GIT mucosal cell. This is the mucosal cell of the GIT. And here we have a poly Ig receptor. Which receptor is there? Here poly Ig receptor. Poly Ig receptor. This poly Ig receptor combines with this dimer, this dimeric IgA, as well as this J chain. Okay. This poly Ig receptor combines with dimeric IgA and J chain, and it gets internalized. After internalization of this whole part, there is an enzymatic cleavage, and after enzymatic cleavage, the secretory IgA is produced. And secretory IgA will have a part of this poly Ig receptor. A part of poly Ig receptor is called as the secretory component. This secretory component or the secretory piece is a part of poly IgA receptor. Okay. So this is the whole story, how the secretory component is being produced. Understood? Yes. Okay. So secretory piece is basically helping in the secretion of dimeric IgA into the lumen and protects secretory IgA and gets attacked by proteases. Let's talk about IgD. We very less know about IgD. Before going to IgD, let's revise the IgA antibody. I am revising the things and telling you the things again and again because this is a like retaining component part. And IgA antibody is basically a local antibody, mucosal antibody, secretory antibody. Okay. LMS you can remember. Okay, LEMS is the your Lambert-Eaton muscular syndrome or dystrophy. You can just remember like that, local, mucosal and secretory. Right, present on large mucosal surfaces, large amount of mucosal surfaces. Most common in case of breast milk and colostrum. IgA is activating the alternate pathway. Two subclasses, IgA and IgA2, A1 and A2. A1 is basically more than A2. So this is the order of serum concentration. IgA exists in two forms. IgA exists in two forms, your serum IgA and the secretory IgA. Serum IgA is valency 2, secretory IgA is valency 4. That is a dimer present on mucosal surfaces, right? And what is the structure of secretory IgA? This is the structure of secretory IgA. Two monomers are there and linked by the J chain, and secretory component or secretory piece is there. This is produced by mucosal epithelial cells. How this is produced? This is a poly IgA receptor, poly Ig receptor or poly IgA receptor, which is internalized and broken down by enzymatic cleavage and secreted in lumen with the piece. A poly IgA receptor is cleaved up, except this piece. This piece is the secretory component, right? And it is helping the secretion of dimeric IgA into the lumen as well as protection against the attack by proteases. Let's talk about IgD. It is not strictly an antibody. It's a monomer having valency 2. Activates alternate pathway again. IgA was activating alternate pathway. This is also activating alternate pathway. And second max carbohydrate content is there in this. Presence of surface of B cells is BCR. Okay, BCR. So you must have been remembering that BCR, we told this like that for one for IgM and one was IgD. Okay, and this was structure BCR, and we had Ig alpha, Ig beta, Ig beta. Here also Ig alpha and Ig beta. And here we had the CD markers, right? What was that? CD19, CD21 and CD81, like that. Okay, this whole structure we told was the BCR. Do you remember the structure BCR? We studied today only, right? Okay, so IgD, repeating it again, monomer, valency 2, activating alternate pathway, second max carbohydrate content, present on surface of B cells. 4 points. Okay, not strictly an antibody. Talking about IgE antibody, IgE, E for extreme. Okay, it has the least serum concentration, shortest T-half, most heat labile of all antibody classes. The most heat labile of all antibody classes, least concentration, shortest T-half. That's why shortest T-half, right? Most heat labile of it gets broken down by heat, and it is known as reagenic antibody or homocytotropic antibody. Reagenic antibody or homocytotropic antibody. Homocytotropic means same cell loving antibody. Okay, it binds to only mast cells and basophils, loves this both cell only. Okay, in humans. So it is mainly reagenic or homocytotropic. Monomer, it is a valency is to maximum carbohydrate content of all classes. Okay, it has the maximum carbohydrate content of all classes. The second maximum was present in the IgD. Do you remember? Second max carbohydrate content IgD. Yes, and the first is the IgM. Okay, binds to the mast cell and basophils. Maximum carbohydrate content. Important role in type 1 hypersensitivity reactions. We will move to the hypersensitivity reactions today only. Okay, in due course of time. Then Fc fragment. What about Fc fragment? Fc fragment means crystallizable fragment. What are the functions? C, 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 C and all C. Functions are all C. Fc fragment C functions. Okay. So Fc fragment, just some points about Fc fragment can be remembered by this mnemonic. Okay, Fc stands for crystallizable. So C means carboxyl end. Fc fragment is present in the carboxyl end, right? Because you know the structure, right? You know the structure. I don't have to repeat it again and again. NS3. This was the COOH, right? So Fc fragment means this part. This crystallizable fragment. This is the Fc, and this is the FAB, and this part was the hinge region, right? We discussed the structure again. Carbohydrate side chain. Fc is the carbohydrate side chain. It can cross the placenta. This Fc fragment can cross the placenta. Complement binding site. CH2 is the complement binding site. CH also determines the class. Okay, determines class, and it is the constant region, and the crystallizable fragment. FAB means antigen binding fragment, and FC is the crystallizable fragment. What are the functions of antibodies? So functions of antibodies can be remembered by this antibody mnemonic only. Antibody. Okay. A for antibody dependent cell mediated cytotoxicity. We discussed about two mechanisms here about an antigen, about the antibody dependent cell mediated cytotoxicity. Do you remember the mechanisms? One was that mechanism in which there was killing due to this antibodies. Fc portion is being bound to that cell, and other things were happening, right? Neutralization of toxins, viruses. Okay, this IgA and IgG cause the neutralization of toxins and viruses. Okay, T is for traveling to placenta. IgG travels to placenta. I is the initiation of classical pathway of complement. Okay, classical pathway is initiated by these antibodies. B is the B cell receptor. T is the T cell receptor. IgM and IgD are B cell receptors. Opsonization. IgG1 and G3 are opsonins. We told that odd numbers, good opsonins, right? Degranulation of mast cells and basophils. IgE cause degranulation of mast cells and basophils. And IgA is yes to a defense colon secretions. In secretory antibody IgA, right? Understood. And about hinge region is rich in proline and cysteine, can bend easily at hinge portion and absent in IgM and IgE. I told that the CH regions are 4 in number, but hinge regions are absent. Do you remember? Yes. So just close your eyes and try to recall all this function of antibodies. So A was the antibody dependent cell mediated cytotoxicity, right? And then N is the neutralization of toxins and viruses. IgA and IgG cause the neutralization of toxins and viruses. A and T means travel to the placenta. Travel to placenta means IgG is basically traveling, crossing the placenta due to its low molecular weight, right? And A and T, I means initiation of classical pathway of complement. The complement pathway, the classical pathway is being initiated by this antibodies, right? And then B is the B cell receptor. IgM, IgD, we know this. And O means opsonization. Opsonization is done by good opsonins, IgG1 and IgG3, right? And D means degranulation of mast cells and basophils by IgE. IgE was called as the reagenic antibody or homocytotropic antibody, right? And Y means yes to a defense colon secretions, IgA. So that completes our antibody. Now let's move on to the next part. Let's move on to another topic which is known as TD and TI antigens and super antigens. Okay, this is a special type of some antigens, biological class of antigens on the basis of inducing antibody formation. So TD antigens means T cell dependent antigens, and TI means T cell independent antigens, okay. So T cell dependent, T cell independent antigens are there, and super antigens we will discuss. So normally all the antigens are T cell dependent, okay. Most of them are T cell dependent, means T cells have to be activated by antigen presentation only. Okay, so APCs are required here. APCs are required for the activation of this T cells. APCs are required, but T cell independent reactants, some antigens are there, like for example your bacterial capsule antigen for bacterial capsule antigen for, you can write bacterial capsule, you just write bacterial capsule, your flagella, and your LPS, lipopolysaccharide. Okay, this don't need T cells and APCs. They directly bind to immunoglobulin receptors on B cells and produce non-specific antibodies. Okay, they produce non-specific antibodies. This is the whole thing about TI and TD antigens. Some important points are there, like dependent antigens. Talking about TI dependent antigens, they are structurally complex proteinaceous antigens, but these are structurally simple antigens. These are simple. T independent are simple antigens. TD antigens are a bit complex antigens, complex structure, you can say. Okay, and T dependent antigens are basically immunogenic, and it doesn't these are not immunogenic, you can write not immunogenic, because these are not requiring the help of this even T cells and APCs. These are directly binding and producing the non-specific antibodies. Memory will be present in this T dependent because the whole normal mechanism is going on now. So memory must be present here. Memory is present over here, but here there is no memory. Here there is no memory. Here there is no antigen presentation. Here the antigen presentation is required over here. Here, and antigen presentation is not required over here, right? And these are the things. And antibodies of all classes can be produced by this. It can generate antibodies of all classes, but this part cannot generate antibodies of all classes. It can generate only IgM and IgG3. Understood? Let's talk about super antigens. Super antigens are the third variety of a biological class of antigens. Okay, the unique feature is that they can activate T cell directly without being processed. Can just if you want to know the definition, these are the things which can activate T cells directly without being processed by antigen presenting cells. These are activating the T cells directly without being processed by antigen presenting cells. Okay, so super antigens are such type of antigens. So various super antigens are there. So basically, what is the receptor of super antigens? So receptor is that variable beta region of TCR. Variable beta region of TCR. You know about the beta region of the TCR, right? You know about the structure of TCR, right? So the variable beta region of TCR, B beta of TCR, appears to be the receptor. B beta of TCR is the receptor, and their non-specific activation of T cells lead to a massive release. The super antigens cause a phenomenon called known as cytokine storm. Very important, cytokine storm, which includes inflammatory mediators and cytokines, interleukin 1, interleukin 6, TNF alpha. Okay, these all are produced very increasedly. Okay, non-specific in activation of T cells is there now. So this are activating T cells directly, means there is non-specific activation of T cells, non-specific activation of T cells. So it is producing the cytokine storm, and this cytokine storm will stimulate the production of, will stimulate the B cells also, and if it stimulates the B cell polyclonally, it will lead to increased secretions of non-specific antibodies, and production of these non-specific antibodies is called as hyper gamma globulinemia. Okay, this is the increased secretion of non-specific antibodies is known as hyper gamma globulinemia. Understood? So many super antigens are present, many super antigens are there. What are the examples of super antigen? Just remember the important ones. Okay, so we have some viral super antigens like Epstein-Barr virus associated super antigen, cytomegalovirus associated super antigen, I am writing SAG for super antigen, rabies nucleocapsid super antigen. Okay, we then we can have fungal one, the Malassezia furfur super antigen. This can also act as the fungus, and these are all viruses, right? Fungus and viruses. And bacterial ones are such present like against the staphylococcal toxin super antigen. Okay, then mycoplasma arthritis mitogen 1 super antigen. This much examples to remember. Okay, this 7 examples are enough for you, and 6 examples. Okay, so EBV associated super antigen, CMV associated super antigen, rabies nucleocapsid super antigen, then you have Malassezia furfur super antigen, staphylococcus toxin super antigen, mycoplasma arthritis mitogen 1 super antigen. Okay, so this is the thing which will cause. Okay, and this super antigen also can cause the super antigens can cause diseases, in humans also. Okay, it can cause diseases in humans. What are the diseases caused by super antigens? So basically we can have toxic shock syndrome, which is caused by this toxic shock syndrome. We can have food poisoning being caused by this food poisoning. Okay, and Kawasaki syndrome, then scalded skin syndrome, Okay, so these all things can be caused by this super antigens. Understood? So super antigens can cause this type of diseases. That completes the super antigen and TD and TI antigens. I hope you understood all these concepts, and what is super antigen is important. It can cause come as a short note question for you. Can activate the T cell directly without being processed by antigen presenting cells. Non-specific activation is the receptor is the B beta of TCR, variable beta chain of TCR. Super antigens can cause a phenomenon. It's called the cytokine storm. The cytokine storm is interleukin 1, 6 and TNF alpha are produced in excess, which will cause the activation of B cells, and these B cells are activated polyclonally. Polyclonal activation is there. Polyclonal activation, this phenomenon is called. Due to which there is increased secretions of non-specific antibodies and causing hyper gamma globulinemias. Okay, hyper gamma globulinemias are these disorders due to excess secretion of non-specific antibodies. Okay, understood. And what are the examples of super antigens? Virus induced EBV associated super antigen, CMV associated super antigen, rabies nucleocapsid super antigen. The fungal one is the Malassezia furfur super antigen, and bacterial one is the staphylococcus toxin super antigen, mycoplasma arthritis mitogen super antigen, staphylococcal and mycoplasma arthritis mitogen 1 super antigen. Understood? And super antigen can cause diseases also, toxic shock syndrome, then food poisoning, Kawasaki and Skalded syndrome. Okay, any other diseases are being caused by the superantigens also, like we can have atopic dermatitis even, okay, by the superantigen. Not important, so don't remember. So that completes our superantigen TI and TD classes. Let's move on to the next part. Let's move on to very important part, antigen antibody reactions. Okay, so we'll first learn about general properties of antigen antibody reactions, then we'll start about conventional immunoassays like precipitation and agglutination reaction. Of course, we had learned about this, about the assay, how can we assay the values, or can say quantities or quantification. Then we'll talk about newer techniques like ELISA. ELISA is very important, enzyme linked immunosorbent assay, and enzyme linked fluorescent assay we'll discuss. Immunofluorescence assay is also there, we'll talk about that. And CLIA, we'll talk about CLIA, chemiluminescence immunoassay, western blot, and some rapid tests, right? So it's a big, very not very big, but very important topic. So what are the general properties? Let's talk about some general properties. Talking about the general properties of antigen antibody reactions. So basically, first one is the specificity. Okay, antigen antibody reaction is basically specific between epitope of an antigen and the corresponding paratope of the antibody. Okay, and the exception to the specificity, the exception I told you about specificity, is the cross specificity. Okay, this is a cross reaction. A similar epitope. We saw about a diagram, right? Remember, where there is a test tube shaped and this type of structure, and this type of structure, it can get bound to this any one of them, right? So these are the examples of cross reactions. So cross reaction are the thing which are exception for specificity. Then it is a non-covalent interaction. Non-covalent interactions, and non-covalent means we had H2 IV, remember, hydrophobic interactions, okay, and hydrogen bondings, then ionic bondings, electrostatic or ionic bonds, and van der Waals interactions, remember. Then third, we have strength, and the strength is influenced by affinity. Strength is influenced by affinity as well as avidity. So affinity, some totals are all non-covalent interactions, okay, between a single epitope with its corresponding paratope is affinity. Affinity is basically how much the antigen wants to get bound to the antibody or vice versa. Avidity is the affinities of all binding sites when multivalent antibody reacts with the complex antigen carrying multiple epitopes. Okay, so basically what happens is that whenever there is a multivalent antibody, multivalent antibody means multiple valencies there, many valencies are there. When it reacts with a complex antigen carrying antigen having with multiple epitopes, so there are a host of reactions, okay, as a host of all affinities. So there is a accumulation of affinities, and wherever there is accumulation of affinities, this is called as the avidity. And avidity is the better indicator, you can say, better indicator of strength of an antigen antibody reaction. Okay, for example, you can have IgM has a low affinity, but then IgG, okay, IgM has a very low affinity than IgG, but IgM is pentavalent, valency is 10. So if the valency is 10 means there will be more antigens, multiple epitopes when it will come. There is a accumulation of efficiencies, and so it has a higher, IgM has a higher avidity. Okay, so it can bind to an antigen, it can bind with antigen more effectively than IgG. Understood? Another thing is that avidity increases with time. Okay, avidity increases with time. So basically this antigen antibody reaction is very important from the diagnostic point of view. Okay, so we can have to assess the quantification of antigen antibody reactions is done basically by things called as immunoassays. Okay, immunoassays, and this immunoassays may be of two types. One may be of antigen detection assay assay, and another one is called as the antibody detection assay. Antibody detection assay. There may be of two types, right? And this may be of and further on the basis of the quantification or qualification, it may be of two types. It may be a qualitative assay, it may be a quantitative assay. Okay, qualitative assay means just we know the qualitative assay. Qualitative assay means we don't know the quantity of this antigen or antibody, we just know if it is present or if it is absent. Exact amount cannot be estimated here. Exact amount not estimated. Exact amount is not estimated. Then we can have quantitative assay. Quantitative assay. In the quantitative assay, here we can estimate the exact amount. Okay, here antibody or antigen titer, you basically say antibody titer. Okay, antibody titer. So measurement of antibody is expressed in terms of titer, TITRE, and here the antibody titer can be measured. Antibody titer can be measured. Okay, on the basis of that we can have a semi-quantitative. Okay, exact quantity here is not known. Exact quantity is not known, but expressed in terms of dilution. Expressed that how much dilution is there. Expressed as so much of dilution. This certain dilution is there. So much of dilution. Okay. Example of semi-quantitative is a Widal test and VDRL test. Widal test and VDRL test are semi-quantitative, and we can have quantitative assays as well. Quantitative means exact quantity here is announced. Okay, we can get to know the exact quantity. Exact quantity means in units like international unit per ml or in mu gram per ml. Okay, examples. What are the examples? Your ELISA. By which process? ELISA, CLIA. Okay, examples. What antibodies are being determined by this? Your anti-COVID antibodies. Anti-COVID. Okay, during COVID times, anti-COVID antibodies or anti-HBs antigen antibodies. Anti-HBs antigen antibodies. Okay, this type of things are determined by this. Understood? Okay, problem is that if the number of antigen or antibody is disproportionate to each other, or if antibody or antigen is present in higher number, then there is no optimal reaction, and often result turns out to be false negative. Okay, to rule out a false negative reaction also, a quantitative test is basically done. So qualitative tests, there in the qualitative assay there may be a false positive present, false positive or false negative may be there. So to rule out this false positive and false negative, quantitative assay must be done to confirm. Okay, understood? Okay, so okay, there is thing that when the, let's say here's the serum, here you take the serum and here antibodies are present. Okay, this when it is diluted by saline, you add the saline, gradually the antibody level will decrease. Antibody level decreases when a fixed quantity of antigen is added to diluted sera. Okay, then there is observed that where the amount of antigen and antibody are equivalent to each other. Okay, there only the antigen antibody reactions are best observed. Means what it say that wherever there is nearly equivalent antigen as well as antibody, wherever there is disproportionation, it is not observed. Wherever there is equivalent, there the antigen antibody reaction is observed best. Okay, understood? And wherever there is a disproportionation, the antigen antibody reactions are least observed. Done? Where the antibodies are excess, the antigen antibody reaction doesn't occur. This is called as prozone phenomenon. Prozone phenomenon. where there is antigen excess, antigen antibody reaction failed to occur. This is called as post-zone phenomenon. Post-zone phenomenon. And this prozone and post-zone phenomenon, these are explained by this hypothesis, is called as the Marag's lattice hypothesis. Understood? Marag's lattice hypothesis. Okay. So it tells that there is a large lattice in the solid state. There is large lattice formed between the antigen and antibody. Wherever there is, so there is, if the levels are equivalent, then the lattice is formed very well. The levels are not equivalent, then it get disrupted. Antibody access means prozone and antigen access means post-zone phenomenon. Okay. Then we are going to the types of antigen antibody reactions. What are the types of antigen antibody reactions? In the types, we have conventional techniques, or you can have the newer techniques. Conventional and newer techniques. So in conventional techniques, we can have many things like precipitation reaction, PPT reaction I am writing, precipitation reaction. We can have agglutination reaction. We can have complement fixation test, we'll read about complements the next. And there is a neutralization reaction, okay. So this is the thing. You can remember CANP for if you want to remember, okay. And we have the newer techniques. What are the newer techniques? We'll read about them also. ELISA, then enzyme linked fluorescent assay. We can have CLIA, chemiluminescent assay, immunofluorescence assay, immunohistochemistry, which you read about in very much in pathology. Then we have rapid tests like flow through assay and all, and Western blotting. ELISA is important, very important for you, okay. So these are the things about conventional techniques as well as newer techniques. This is the overview. Let's move on and study about each one of them in detail. So precipitation reaction, you know, when a soluble antigen reacts with antibody in the presence of an optimal temperature, pH and electrolyte, a antigen antibody complex is formed, okay, where there is a formation of insoluble floccules, okay, or it may form a precipitation band or floccule. It may form a band or it may form a floccule. Main thing is that when a soluble antigen must act, antigen must be soluble, and in the presence of optimal temperature must be optimal, pH must be optimal, electrolytes must be there, okay. It leads to formation of a antigen antibody complex. Understood? And the result of if electric current is there, we call it as immunoelectrophoresis here, okay. What are the clinical applications? It was earlier, it was very widely used for serological tests, but now new techniques are there which are relatively more cost effective, simpler, as well as very this thing, okay, not commonly used nowadays, but some applications are there, like slide flocculation test we do for a syphilis. Slide flocculation tests for syphilis. Slide flocculation test for syphilis, it induces serodiagnosis of syphilis, okay. Syphilis is being a sexually transmitted infection by Treponema pallidum, okay. So a drop of antigen is mixed with antibody on a slide, and floccules are formed here, okay. And many tests are there, like VRDL and RPR tests. Then for diphtheria toxin, we have another test which is called the LX gel precipitation test. LX gel precipitation test, which we do for diphtheria toxin, okay. Diphtheria toxin. So a strain of Corynebacterium diphtheriae is isolated and streaked to a medium containing filter paper. If the strain is toxigenic, okay, it produces the toxin, and it's form arrow-shaped precipitation band, okay. If the toxigenic strain, there is a toxigenic strain, it meets with antitoxin and produces arrow-shaped, this type of arrow-shaped precipitation band is being formed, okay. So this is about precipitation reactions. Let's talk about the agglutination reactions now. Agglutination reactions are important. Here the antigen is not soluble. Here we are taking the particulate or insoluble antigen. Insoluble antigen is taken, then mixed with the antibody in the presence of electrolytes at suitable temperature and pH. The particles are clumped. There is a clumping. Okay, and just remember, in presence of sensitivity is more than this precipitation reaction, more sensitivity, and clumps are better visualized, better visualization also. On the basis of this floccules, here floccules and bands are present, but here there is the better visualization due to formation of clumps. Okay, and agglutination reactions are classified on the basis of your classified as direct. It may be direct, it may be indirect or passive, or it may be reverse passive. Okay, so direct agglutination reaction, or if you want to just sometimes these questions are also asked, what are the different types of agglutination reaction. Agglutination reaction is a bit important. Precipitation reaction you can just leave out, but agglutination reaction is a bit important, so I'll be discussing it in a bit detail. Okay, then we'll come to the complement fixation test and utilization. Very short topics are there. Then ELISA is again a big topic. Okay, so let's start about this agglutination reaction. First we'll discuss the direct agglutination. I'm writing AGGL for agglutination reaction. Direct means the antigen directly reacts with the antibody. Okay, agglutinates directly. So then we have the slide agglutination. Okay, slide agglutination, what is done here? Let's say we have a bacterial colony is there. It's mixed with a drop of saline to form a smooth white suspension is formed. To it you add anti-serum, plus it is shaken or rotated manually for few seconds, and if there is a clumping with clearing of suspension, then it is called as a positive result. If unchanged suspension, it is called as a negative result. This is called slide agglutination. Then we are taking tube agglutination. Here antibody titer can be estimated as highest dilution of serum. Okay, it produces a visible agglutination reaction. Okay, so a fixed volume of you can take a serum sample. Okay, this is a serum sample. Plus a fixed volume of particulate antigen suspension. This is a antigen suspension. Equal volumes are there. Let's say V and V. Equal volumes are added. Okay. If the bottom of test tube will have clumping, when these are added, there is a clumping at bottom of test tube with clearing of supernatant. This is called as a positive test, and if there is no nothing, then it is a negative test. Okay, so where these are used, this slide agglutination reaction and this tube agglutination reaction. This slide agglutination, do you have an example for this? You must have done this in first year, I am sure. It is used in the blood grouping and cross matching. Slide agglutination. What are the tube agglutination methods done? In the serological diagnosis of diseases. For example, your Widal test for typhoid, Widal test. Antibodies against H and O antigens of Salmonella typhi. Okay. So this H antigens are form loose clumps. Okay, this forms loose clumps, and this O antigens, these are forming chalk white dense deposits, chalk white dense deposits. Okay, H antigens form loose clumping, and this O antigens form chalk white dense deposits. Okay, and you can also do tube agglutination in the test for brucellosis. Okay, standard agglutination test is called. Okay, Coombs test is one of the important example. Coombs test. Okay, then we can have some tests like cold agglutination test and Wheel-Felix reaction for typhus fever. Okay, did you understand what I am saying? So basically blood grouping and cross matching are the examples of slide agglutination, and Coombs test, cold agglutination, Wheel-Felix are an example of your tube agglutination. Okay, so after this let's move on to the microscopic agglutination. Microscopic agglutination, just one line. Microscopic agglutination, AGGL, is done for, this is called MAT, microscopic agglutination test, is done for leptospirosis. Okay, is done micro titer plate and visualized under the microscope for agglutination. Okay, so these are the examples of tube agglutination, and slide agglutination are the examples of direct agglutination reaction. Let's move on to the indirect reaction. Indirect or passive agglutination reaction. So this passive agglutination is for antibody detection. Okay, and here an attempt is made to convert the precipitation reaction into an agglutination reaction. Okay, so basically what is being done is that the antigen on the surface of a carrier molecule, like we are coating the antigen with some particles. Likewise, we had seen the latex agglutination test. Okay, so here we are coating antigen with latex or RBCs or your bentonite. Okay, bentonite, so that the antibody binds to the coated antigen. Okay, so antibodies are basically binding to the coated antigen like this, and there is a clumping. And agglutination reaction takes place. Antigen antibody reaction takes place. So basically we are coating the antibody with some latex particles or bentonite or RBC. Okay, so we have many things like passive agglutination tests. Many things are there, like indirect hemagglutination test, IHA. Okay, or RBCs are used as carrier molecules. Then we have the latex agglutination test. We talked about latex agglutination test, right? Okay, so here polystyrene latex particles are used as carrier molecules. Like if it is a antigen, we are coating it with latex. This is latex, polystyrene latex, which is of 0.8 to 1 μm in diameter. Okay, this is the latex particles which we are coating it, and from the patient's serum plus serum has antibodies. Serum will have antibodies like this. This antibodies will be there. This antibodies will come like this and bind to this and serum, and this is rotated also. Positive if there is a clumps forming. Okay, it's the positive result, and it is used. What is the use? Latex agglutination test used for the detection of ASO. What is ASO titer basically? So ASO stands for anti-streptolysin O antibody. So anti-streptolysin O antibody is being detected by this ASO titer. Understood? Then we have the reverse passive agglutination test. Okay, reverse passive agglutination test. This is for antigen detection. So in this reverse passive agglutination test, the antibody is coated with carrier molecule which detects antigen in patient's serum. Okay, so antibody here, the antibody is coated with coated on a carrier molecule. So if this is a carrier molecule, here the antibodies are coated like this. Okay, and it is used for detection of antigen. So antigen, when it will bind, will cause a reaction, and this will be there. Okay, so here reverse passive hemagglutination assay RPHA is also done, and the latex agglutination test for antigen detection is also used. So just remember this: latex agglutination test for antigen detection is done for CRP, is done for detection of CRP, that is C reactive protein. You know the this CRP, rheumatoid arthritis antigen, okay, rheumatoid arthritis factor, capsular antigen in your CSF, okay, capsular antigen in CSF, like this: CRP, RA, and capsular antigen CSF. This latex agglutination test for antigen detection, important, is done. Okay, and then we have heme agglutination tests also. Heme agglutination test. Heme agglutination tests, we can have two types: direct or indirect. This indirect is now obsolete. Direct means what happens: the serum antibodies agglutinate with surface antigen of heme agglutination in blood. Blood means RBCs to form a MAT. MAT means a antigen antibody complex. Okay, form a MAT. Examples are there like Paul Brunel test, employ CIP blood to detect Epstein-Barr virus, okay, antibodies. Then we have cold agglutination test, blood grouping. Okay, you can remember like this. And you can remember PCB, Paul Brunel test, and cold agglutination, this cold agglutination test, CAT, and you have Coombs test also, and BB is for blood PCB. Understood? So these are the direct hemagglutination tests. Understood? Let's next come to your complement fixation test, CFT. Next was the after agglutination test. We discussed all about agglutination test. Now let's move on to the next part. Okay, so before coming to complement fixation test, let's quickly go through hemagglutination tests or whole total agglutination tests. Let's read them again. Agglutination means a particular insoluble antigen is there. There's clumping, increased sensitivity from precipitation reaction, better visualization, clumps are better visible, maybe direct, indirect, or reverse passive. Okay, and for example, direct, let's come to direct. Direct means direct antibody and antigen reaction. We have slide agglutination, just ignore the noises. Slide agglutination, where bacterial colony plus saline water, there's a suspension is made and antiserum is added, shaken, rotated. Clumping with clearing of suspension means positive, unchanged means negative. Example is blood grouping and cross matching. Tube agglutination, serological diagnosis of disease like Widal test, and here H and O antigen as Salmonella typhus acid, where there is loose clumping in H antigen and O antigen, there is chalk white dense deposits, right? So serum sample is taken and antigen suspension is made equal volume mixed, and wherever there is clumping, there is a settling down at the bottom of the test tube. Sorry. Clearing of the suspension. Coombs test and cold agglutination wheel Felix for typhus fever is the example. Microscopic agglutination test, MAT, is done for leptospirosis, important. Indirect or passive agglutination is for antibody detection, not antigen detection, antibody detection. Here the antigen is coated by an antibody coating antigen with latex or RBC or bentonite, okay, and these are recognized by the antibodies. Like polystyrene latex particles are coated by this. For example, ASO titer. ASO is anti-streptolysin O antibody titer. Reverse passive agglutination test is for antigen detection. Here a carrier molecule is there which is coated with this antibodies, and the antigens are detected. So latest agglutination test for antigen detection is for CRP, RA, rheumatoid arthritis factor, and capsular antigen is CSF. Then we have heme agglutination test for indirect and direct. Indirect is obsolete now. Direct for serum antigen plus surface antigen, serum antibody plus surface antigen of RBCs, where there is a mat formation. Like Powell-Brunel test for SIB blood to detect Epstein-Barr virus antibody. Okay, SIB blood to detect Epstein-Barr virus antibody. Cold agglutination test and blood grouping. Okay, PCB. P is for Powell-Brunel test, C is for cold agglutination and Coombs test, and B is for blood grouping. Okay. So that completes our agglutination test. Next we are into the complement fixation tests. Complement fixation tests. Complement fixation tests, or CFT, is detecting antibodies in patient's serum after they are capable of fixing with complements. It was once very popular, now it is obsolete, right? So CFT was widely used for detection of complement fixing in antibodies, for detection of complement fixing antibodies in case of rickets. You can remember something like RAM. Okay, just let me RAM. Okay, RAM means your R for rickettsia. A is for arbovirus infections, and M is for mycoplasma. Okay, RAM. And another example of chlamydia is also there. Okay, so chlamydia is also there. Just a minute. So you can remember this chlamydia also by this C, complement fixation, the CFT, chlamydia. Okay, and complements are also used for various serological tests, such as your for serological tests like treponema pallidum immobilization test, TPIT is treponema pallidum immobilization tests. Okay, for syphilis. Just remember one example. This is not much important. And then we have the neutralization tests. Okay, neutralization tests. So we have in neutralization tests, we have viral neutralization test, presence of neutralizing antibody in patient's serum. When the serum is mixed with live viral suspension and poured into a cell line, then specific serum antibody will neutralize it. Okay, and this virus will be unable to affect a cell line. Viral neutralization test. Then we have plague inhibition test, viral neutralization, plague inhibition for bacteriophage. Okay, and then we have toxin antitoxin neutralization test, antitoxin neutralization test. Okay, toxin antitoxin neutralization test. For example, you have SCHICK, sick test, and Naegler's reaction, NAGLERS, Naegler's reaction. Sick test and Naegler's reaction are two toxin antitoxin neutralization tests. So sick test is a diphtheria, for diphtheria. Sick test is for diphtheria, and Naegler's reaction is for alpha toxin of C. perfringens, Clostridium perfringens. Okay, so Cartoon Network you must have known, CN. Okay, and this thing you can use to remember this. Okay, neutralization test. So we have three type of tests. Viral neutralization test. Okay, it will make the virus incompetent to affect the cell line. Then we have plague inhibition test. Plague inhibition test is for bacteriophage. Okay, and then we have toxin antitoxin neutralization test. We have two examples: sick test and Naegler's reaction. Sick test is for diphtheria, and Naegler's reaction is for Clostridium perfringens. Right? Understood. And we have another example, also ASO test. Okay, earlier it was used, and neutralization method also, this ASO test was used. Okay, you can write ASO test for this. Understood? This one. Then we have some newer techniques. These are the conventional techniques for this part, and then we have some newer techniques. Newer techniques. Newer techniques are important. Newer techniques use a detector molecule to label an antigen or antibody, which in turn detects the corresponding antigen or antibody by producing a visible chemical effect. Okay, so first one is the ELISA. ELISA. Okay, let's move on to the ELISA first, then we'll discuss the other things. Other things are less important. ELISA is more important. Okay, let's discuss the ELISA from this PDF. ELISA means enzyme linked immunosorbent assay, detection of antigen and antibodies. Okay, immunosorbent means it depends on the antigen antibody reaction. Okay, can be used for quantitative and qualitative analysis of antigen and antibody. Both analysis are there. Antigens, we know proteins, peptides, hormones, bacterial or viral antigens it may be. Immunoassays, basically radio immunoassay, we have ELISA and CLIA. Three types of assays are there. Radio immunoassays, we have radiation hazards and shorter shelf life of reagents. Disadvantage is there, so we use ELISA. ELISA have no radiation, any radioactive reagents are not employed, right? So we have no radiation hazards. Shelf life also is longer and easy automation. And the method is very easy. Okay, a bit costly, but CLIA is chemiluminescent immunoassay. We'll discuss about that later. Uses of ELISA is for estimation of hormones like T3, T4, TSH. The thyroid hormones are estimated by ELISA only. Thyroid hormones are estimated by ELISA, T3, T4, TSH. And used for detection of antibody against HIV virus. Used for SARS-CoV-2 infection detection also. Okay, so SARS-CoV-2, the COVID, employed the ELISA technique also. CLIA is ultra sensitive, ultra specific and easy automation. Partly has replaced ELISA. Okay, the sensitivity of CLIA is more than ELISA. The specificity of CLIA is also more than ELISA, and automation is easy. So what is the definition of ELISA? Immunochemical method which employs the use of antigen antibody reaction in order to detect antigen or antibody. Very simple definition, you all can understand. The antigen is adsorbed on a solid phase support. Okay, the antigen is absorbed on a solid, adsorbed, important line is adsorbed, not absorbed. Adsorption is different thing, right? A solid phase support. What does the solid phase support use? PVC or polystyrene. They're fixed on the antigens are fixed on the PVC and polystyrene, and the phenomena is known as passive absorption. Okay, passive absorption. Understood? So basically here in this plate, this part is made up of PVC and polystyrene. This surface is basically made up of PVC or polystyrene, where the antigens are adsorbed or fixed by passive absorption method, and there is a direct capture in the direct ELISA, or there is a indirect capture or indirect ELISA. It may be that this PVC or polystyrene may be used to fix the antibody. It may be used to fix the antigen, or it may be used to fix the antibody, right? So these are the antigens. It may be fixed, or it may have to fix the antibody depending on the type of ELISA, right? What are the enzymes involved in the ELISA? We have the most common is the horseradish peroxidase, HRP enzyme, ALP enzyme, beta glucosidase, glucose oxidase, and ACHE. Okay, horseradish peroxidase is ALP, beta glucosidase, glucose oxidase, and ACHE. ACHE is the acetylcholine esterase, right? HRP, ALP, beta glucosidase, glucose oxidase, or ACHE. Linking substances. The substances used to link, okay, between the antigen and antibody is the glutaraldehyde. Okay, others are periodate and melamide. Glutaraldehyde is the most common, and others are periodate and melamide. So I have four types of ELISA basically, and other ELISAs are also there. I will tell about them. We have some MAC ELISA and ELISA lot and all. Okay, I will tell about them. Don't worry. Four types of ELISA are basically there based on detection: direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. Okay, direct, indirect, sandwich, and competitive. Just understand what I am saying. So direct ELISA is for detection of antigen. Okay, indirect is for antibody detection. Sandwich for antigen. Okay, and competitive is also for antigen. Only indirect ELISA is for detection of antibody. Understood? Only indirect ELISA is for detection of antibody. These diagrams you must draw in your exams, so that the examiner is impressed by your diagrammatic skills and all. Okay, so let's discuss first, then we'll start. So direct ELISA, in which we have a PVC or polystyrene surface here. Okay, the antigen is fixed. Antigen is fixed here, and we put an antibody as a primary antibody. Okay, the antibody is linked with an enzyme. Okay, the antibody is linked with an enzyme which converts the substrate to a colorful product. So it is a chromogenic thing. Okay, so substrate, what are the substrates? We may use TMB, tetramethyl benzidine, tetramethylbenzidine, aminosalicylic acid, or OPD, orthophenyldiamine, or diammonium salt. Okay, you can use tetramethylbenzidine, which is the most common, TMB, tetramethylbenzidine, or aminosalicylic acid, or OPD, orthophenyldiamine. Okay, these are the substrates. So substrate is converted to the product, and this product we can quantify, detect, right? This is the direct ELISA. Understood? This is the direct. Did you understand the direct ELISA? Yes. Okay. In the well, a antigen is put, the primary antibody linked with enzyme is put, and the substrate chromogen, the chromogen will change the color, right? After that, indirect ELISA. Indirect ELISA is basically employing primary antibody as well as secondary antibody, which will be attached to the Fc portion of primary antibody. Okay, so here the same antigen is fixed on the PVC or polystyrene surface, and the antibody of interest is now attached, antibody of interest, and this is also linked by secondary antibodies, the FAB part of secondary antibody linked to the Fc part of primary antibody, and this will have the enzymes. So there is an amplification. Look at this, here one enzyme was there, here two enzymes will be there, means there will be amplification, more product, there will be more product, substrate to product, more, okay, more product. So there is an amplification of signals. So signal amplification is possible here. So it is used to detect the antibody against HIV. Very important, just remember the example. Then we have another sandwich ELISA. Sandwich ELISA is a very unique type of thing. It is also used to detect the antigen. Okay, here we are detecting antibody of interest, so this is used to detect the antibody, but sandwich ELISA is sandwich, the antigen sandwich between two antibodies. Here the antigen is not captured in the sandwich ELISA, antigen is not captured, a antibody is captured here, so in this polystyrene antibody is captured. This is the antigen of interest, for example TSH. This sandwich ELISA is employed for the assay of TSH also. Okay, and this is let's say is antigen of interest, TSH, and another antibody linked with primary antibody first comes, then it will be linked with the secondary antibodies. Okay, so there is a more amplification of signals. Okay, the signals are employed to a very higher extent, and it is now replaced by avidin biotin chemistry also. Okay, means the antibody is linked with avidin and streptavidin, okay, and the substrate to product conversion occurs here. Okay, then we have the competitive ELISA, where the small antigen is used. Okay, in competitive ELISA, this is also known as inhibition ELISA, to determine the concentration of T3 and T4. T3 T4 concentration for determination we use competitive ELISA. For TSH we use this ELISA. Which one? Sandwich ELISA, right? For TSH we use sandwich ELISA, you can see. Okay, so what happens in competitive ELISA means, first of all we take this surface. Okay, this PVC surface is there. Here we capture all the antibodies are captured. Okay, antibodies are captured, and here these are enzyme linked. Okay, these are enzyme linked, and here a conjugate antigen is there. Here a conjugate antigen which is labeled with enzyme is there. Okay, this part. Basically we are capturing the serum antigens, and the conjugate antigen labeled with enzyme is put now. There is a competition between the serum antigen to bind and the conjugate antigen. How much competition is being determined by this color intensity. After it is washed, this whole system is washed, and after that the concentration T3 and T4 I told. So this concentration of T3 will be inversely proportional to the color intensity. More the color, there is less T3, because there is more competition, right? More competition means there will be more color intensity, because enzymes are more linked. Okay, more conversion of the substrate to the chromogenic substrate, right? So concentration of T3 is inversely proportional to the color intensity. Substrate color intensity inversely proportional to the concentration of T3, right? So this is about your sandwich ELISA. Understood? And again we have other types of ELISA also. If you want to just know, other types of ELISA are there. One is the IgM antibody capture ELISA, MAC ELISA. Okay, and other type is the MAC ELISA. MAC ELISA is IgM antibody capture ELISA. Okay, basically this is the type of sandwich ELISA only. Type of sandwich ELISA. This is a type of sandwich ELISA. Okay, it is used for detection of dengue, Japanese encephalitis, JE I am writing, okay, and leptospirosis also, leptospirosis. Conventional method used for leptospirosis was MAT, agglutination test, microscopic agglutination test, right? And West Nile virus, all this, okay. It is based on the capturing primary IgM antibody on the microtiter plate pre-coated with the anti-human IgM antibody, okay. That what it means is that this is the surface, okay, and here the captured ones are the anti-IgM antibodies, okay. These are the anti-human IgM antibody, okay, understood. Then we add the recombinant antigen, for example our dengue antigen. Here the dengue antigen will be added like this and like this, okay, dengue antigen. Then we are capturing the primary antibody. I had drawn in that orange color, so I am maintaining the color code from the previous discussion. You can see orange color was used, so let's draw it with orange. This is the primary antibody which will be done, okay. And then another secondary antibody will be used. The secondary antibody, this is a secondary antibody, I'm drawing 2, okay, linked with enzyme, all linked with enzyme. Enzyme will be product to substrate, substrate will be formed in each case, so there is an amplification, right? Understood. And more amplification, you use to get more amplification, we use avidin or streptavidin biotin system, for more amplification. Understood. So these are the things about MAC ELISA and IgM antibody capture ELISA or the MAC ELISA, right? Next thing, another test is there. If you want to know, a test is there which is called as your ELISpot test. ELISpot test, okay. ELISpot test, what is it basically? Here, ELISpot test is a modification of ELISA, okay. It is a modification of ELISA, which we use for quantitative detection of cells producing antibodies like plasma cells or cells, just a minute, or cells producing cytokines, for example lymphocytes and macrophages. Understood? Done? Okay. Next we have another thing which is known as IgG avidity ELISA, avidity term, I taught about that. IgG avidity ELISA. So IgG avidity ELISA is used to differentiate recent infection from past infection, to differentiate between recent and past infections, okay, to differentiate between recent infection from the past infection, okay. So recent infection and past infection, how can you differentiate? IgM will be present. Past infection means gone by. IgG will be present, right? And this situation where both the antibodies are present, IgG avidity test is used to differentiate between them. Okay, avidity of an antibody is the basically the how firmly it is attached to the antigen, right? Avidity reflects the maturity of antibody. Okay, avidity is reflecting the maturity of antibody with time. Okay, so low avidity IgG means recent infection. Okay, high avidity IgG will indicate a what infection? Past infection. Okay, this much you must know. And examples for detection of, for diagnosis, for I'm not DX, for diagnosis of many things. For diagnosis of what things? Infections like rubella, cytomegalovirus CMV, BZV, toxoplasmosis, EBV, HIV, all viruses you can write. Okay, West Nile virus also. Understood? So this is the IgG avidity ELISA. Advantages of ELISA, you know, it economical, 2-3 hours per performing the assay. ELISA has high sensitivity also, but chemiluminescence assay has more sensitivity. Disadvantages, what are disadvantages is that it basically rapid test will take many 10 to 15 minutes time, but ELISA will take 2-3 hours, so comparatively more time, and expensive instruments are used. Okay, and the instruments are a little bit expensive, right? So this is the thing. And then we have other assays, okay, which are enzyme linked fluorescent assay, immunofluorescence assay, and CLIA, chemiluminescence assay, and rapid test. Let's read about that 4, and then we'll finish the antigen antibody reactions. Of course, it is a like bigger topic, so it is taking time as expected. Okay, and then we'll move to the next part. Okay, let's move on to the next part first. Okay, so let's talk about alpha and IFA. Okay, so enzyme linked fluorescent assay is basically enzyme linked ELISA in 2 different ways, like automated system. Okay, ELISA is done a bit manually, semi automatically you can say, but here we have a completely automated system. Okay, and here fluorometric systems are used. Okay, fluorometric methods are used. Okay, example are VIDAS, VIDAS, and mini VIDAS systems are used. Okay, and it is more easy, friendly, eco-friendly, you can say, less contamination chances, and more sensitive, and more specific, right? What are the disadvantages? It is expensive, very expensive, and a limited number of parameters can be tested. Okay, so for example, it can be used for detection of hepatitis viruses, HIV, antibody to torch infections in pregnant women. We'll read about torch infections when you read systemic microbiology, torch infections. Okay, and dengue, SARS-CoV-2 also important, SARS-CoV-2. Other detection of tumor markers. Okay, for example, tumor markers, or detection of cardiac markers. Okay, all of this. Understood? And IFA is immunofluorescence assay. It is similar to ELISA, but fluorescent dye is used. Fluorescence dye is used to label antibody. Fluorescence dye is used to label the antibody. It detects the cell surface antigens. Okay, unlike ELISA only detects the free antigens, but this is also used for detection of cell surface antigens also. Surface antigens also used for detection of cell surface antigens also. Understood? And principle is that fluorescence means your glowing nature. Okay, visible lights are obtained. Okay, and fluorescence compound which is commonly used, the fluorescence compound which is commonly used is your fluorosin isothiocyanate. Fluorosin isothiocyanate, FITC. FITC is commonly used, right? What are the types? Direct and indirect. Two types of immunoassays are there. One is a direct, which is directly tagged with that antigen. Fluorescent dye is added and washed, viewed under fluorescence microscopy. And indirect. Indirect means antibodies are detected in the samples. Okay, here direct is used for your surface antigens. Okay, antigens are detected, right? And here indirect, the antibodies are detected. Antibodies are detected. Serum containing primary antibodies added to the slide and washed to remove that and all. Okay. Many the applications are there, like detection of autoantibodies, anti-nuclear antibodies, or rabies antigen. Okay, many things are there. Flow cytometry, you know about. There is an antigen which is fixed. Flow cytometry. Just let me tell you in a little bit detail. Okay, as it is a newer method, so I'm just covering it for completion's sake. Flow cytometry is laser based technique actually. Okay, here in the surface, okay, in a surface, this antigen is fixed, and then we are adding a primary antibodies. Primary antibody has a fluorescent label. Okay, this primary antibody has a glowing label. Let me draw it with this type of green. So as you remember, the fluorescent label present, there is a glowing nature. Okay, and when this anti-human IgG conjugated with fluorescein is added, okay, and visualized with the as the cells are separated as they pass through the laser beam. Okay, so they are used for assessing the parameters of the cell. Okay, assessing the parameters of cell, such as cell count, cell counting. Okay, used for cell counting, or it may be used for DNA or RNA content determination. DNA RNA content determination. Okay, or it may be used for your analysis, any cell analysis of any shape, size, shape of cell. Okay, in HIV infected person, CD4 T cell counting is done by this method. CD4 cell counting is by this method. Okay, all of this. Then we come to CLIA, or chemiluminescent immunoassay. CLIA is refers to the emission of light as a result of chemical reaction. Okay, so there is a chemical reaction occurring. There is a chemical reaction occurring, and chromogenic substrate is there is no chromogenic substrate. No chromogenic substrate. We use chemiluminescent substrates. Chemiluminescent substrates used. Okay, chemiluminescent substrates used like luminol and acridinium ester. Luminol and acridinium ester. Luminol and acridinium ester. Okay, which generate a light. Okay, these are called as luxogenic. Luxogenic means luminance generating. Okay, luxogenic. Understood. And CLIA is 10 times more sensitive than ELISA. Okay, about 200 folds more in improvement. Understood. But CLIA has a very limited application in diagnostic microbiology, like antigen, torch infections, HIV, and all these are used. Okay, for estimation. So that's why. Then we have the Western blotting. Many blottings are there. You can remember SNOWDROP for remembering the blotting. Snowdrop. So southern blotting, as for southern blotting, is for your DNA, and northern blotting is for your RNA. O is O. Then Western blotting is for your proteins. Okay, so Western blotting is determination of specific proteins, antibodies in a sample containing a mixture of antibodies when targeted against different antigens. Okay, a specific antibody is detected. A specific antibody is detected from a combination different antibodies against different antigens. Okay, this is the thing. So what are the steps, basic steps? You must know briefly, not detailed. Okay, separation of antigen mixture into different fragments. Okay, the antigen mixture is separated. First one is the separation, and separation is done by gel electrophoresis. Gel electrophoresis separation. Then we have the transfer onto nitrocellulose membrane. Okay, and then we do enzyme immunoassay. Enzyme immuno. Okay, understood? Done. Okay, then some rapid tests are available. Rapid tests are revolutionary diagnostic of this infectious disease, very simple to perform. Okay, about rapid test, if rapid test kits are there sometimes. Okay, rapid test kits and other ICT kits are used. Okay, this is principle of ICT kits, means many other like immunochromatographic test. ICT is immunochromatographic tests based on lateral flow technique. It is used in diagnostic microbiology most commonly. So rapid test, if I talk about rapid tests, okay, are very less minute, 10 to 20 minutes they take. Okay, these are called the point of care tests. Okay, because POC tests, point of care tests, because this test can be performed independently of laboratory. Though they don't require any specific or experienced individual, and can be done in any laboratories. Okay, just a minute. And rapid tests are available for many diseases like malaria. Malaria you must have seen, maybe malaria. Okay, for hepatitis B, for hepatitis C, for HIV, leptospirosis, for syphilis. Okay, many rapid tests are available, and two mechanisms are there for rapid tests. One is called as the lateral flow assay mechanism, lateral flow assay, and flow through assay. This is called as the ICT, your immunochromatographic test. Okay, understood. Principle, what is the principle? This test system consists of a nitrocellulose membrane and adsorbent pad. Okay, this is coated by this nitrocellulose membrane. This is this is nitrocellulose membrane. This is nitrocellulose membrane, which is coated by two lines. Okay, which consists of two line. Understood? A test line coated with monoclonal antibody. This is a test line. Let's say this is a test line coated with a monoclonal antibody. Understood? And the control line coated with anti-species immunoglobulin. Control line containing an anti-species immunoglobulin. Okay, and antibody against the targeted antigen labeled with chromatogenic marker is added, infiltrated into the sample window. Okay, means a specific antibody tagged with colloidal gold or silver is added basically. Okay, so antibody gold or silver. Okay, this is added, this is infiltrated against the specific antigen. Okay, against the test antigen. If this is the test antigen, this is the control antigen, you can say. This is the test line. Okay, both antigen specific antibody colloidal gold complex and the free colloidal gold level antibody move laterally against the cellulose membrane. Okay, so this is the ICT test. Test band here. In the test band, there is a formation of a colored band here. Colored band. Okay, and the control band is also colored. This is always colored, but here the color depends upon if the antigen and antibody are complementary to each other, or the detection is positive, the test is positive, then it will show color. The test is negative, then it will remain colorless. This is the method of lateral flow, because the flow is laterally, okay, towards this side, laterally. Against flow through assay, a protein A is used to label instead of gold antigen. We use protein A, and the sample flows vertically. Understood? So here there is a this concept of test dots and control dots. Okay, if the test is positive, then pinkish purple dots are given. Pinkish purple dots. Okay, the control dots is always giving a pinkish purple. If test is negative, then colorless. Here are always pink, pinkish, right? So this completes your antigen antibody reaction in toto. Okay, these are all about the antigen antibody reaction, and next topic will be your complements. Next we'll move to the complement system. Then next, okay, complement system. Okay, so let us discuss the complement system now. Very important topic. Group of proteins belonging to innate immunity, very important, innate immunity, which help in the acquired immune response and in inflammation. Okay, this is the complement system. And these proteins are glycoproteins produced by the liver. Liver is the main site, hepatocytes, and also produced by blood and tissue, blood monocytes and tissue macrophages also form this glycoproteins, and epithelial cells of GIT and GUT. Okay, GIT and GUT, genitourinary and gastrointestinal, right? Complement proteins are 5% of total serum globulin fraction. They are inactive proteins or prozymes or zymozymes, which by proteolytic cleavage the active sites are exposed. Three pathways are there basically, and they belong to active protein, become active protein and cause the killing of the microbe. So they are inactive proteins. And how they are activated? We have three pathways of activation: classical pathway, alternative pathway, lectin pathway. So we have to read these three pathways in detail. Any pathway can be asked as a question in your exam, right? And it is very important from diagnostic point of view also. We'll see about complement related disorders as well, right? So the most important one, that's the classical pathway. Let's discuss the classical pathway first. So basically what happens is that it is a recently discovered pathway. You might be wondering that classical means it is from a long period of time. No, it is a recently discovered pathway activated by antigen antibody complexes or immune complexes, not by any antigen or antibody alone. Okay, activated by immune complexes. So what happens is that first this is the pathogen. Okay, this is the pathogen, and this is our antibody, right? So this antibody has a CH2 region. This CH2, this is the domain which is infolded. Okay, it is not outfolded, it is infolded like this, right? So CH2, when the pathogen binds to here, the CH2 will open up. The CH2 open up. This opening of CH2, provides binding of C1 complement. C1 complement is a peculiar structure. There's C1q, C1r and C1s. Okay, so three are bound like this. Okay, and the C1s has esterase activity. S for S, esterase. The S pronunciation is that right? Okay. So this is C1, the structure of C1 you can see in the electron microscopy picture is there, or this is the structure. You might have, you don't have to draw the structure basically, you can just know the structure. So one C1 molecule is made up of one C1q, two C1r and two C1s. Okay, C1q is only one number, two C1r and two C1s. This is the C1 complex, which is also written like this. It is also written like C1qr2s2. C1qr2s2, right? This is the whole thing of C1, and this subunits are stabilized by calcium ions. So calcium ion is a necessary factor for classical pathway. Then step 2 is that C4 comes after C1. C4 comes, and C4 also, C4b it divides into two subunits. C4 comes and binds. C4b is binding and C4a will be leaving. It is written like this: C14b. C1 is bound, C4b is bound, okay, and a bar above it, okay, C14b. After that, step 3, according to QBC immunology, C2 comes, but C2a only binds. C2a only binds and C2b's go away, okay, C2b goes away. So here C1q, C1r, C1s, C4b is binding, C2a is binding, not b, C2a is binding, okay. This is called as C14b2a, C14b2a, okay. Whatever is binding, you just write it. This is called as C3 convertase, C3 convertase. But according to Warren-Levinson textbook, this C3 convertase is C14b2b. According to Warren-Levinson, this C2 this C2b binds and C2a goes away according to Warren-Levinson textbook. But we follow the QBC immunology more commonly. So, and Apurva Sastry also writes that C2a only binds and C2b goes away. So I am following one of them, okay. So C3 convertase is C1b, C14b2a, C14b2a. After that, step 4. Step 4, what happens? C3 will come. C3 convertase was there, right? C3. So C3 will come, C3b binds and C3a goes away, right? So what is the sequence? Sequence is 1, 4, 2, 3, where 1 was completely bound, 4b was bound, 2a was bound, 3b was bound, okay. So this is called the C5 convertase, C1b, C1, 4b, 2a, 3b. This is the C5 convertase, C5 convertase, okay. So then C5 will come, obviously. C5 comes, C5b doesn't bind here. There is a catch here, that C5 won't bind back to the complex. It binds with other proteins and conspires, means it binds with its army. C5b binds with other proteins like C6, C7, C8 and some molecules of C9, okay. So it binds with other proteins like C6, C7, C8 and other molecules of C9. It's a cylindrical proteins. C5a goes away, but C5b doesn't bind like other complements, right? When they bind together, the C5b, C6, C7, C8, C9 molecules, this is called MAC, membrane attack complex. MAC is the membrane attack complex. This membrane attack complex will insert holes in the membrane, okay. This will insert in the membrane and drills hole in the membrane, which will cause the killing of the microbe. This MAC finds it difficult to attack gram-positive because gram-positive has a thick membrane, right? So it will be more susceptible. It will very easily kill the gram-negative than the gram-positive. And this classical pathway is not activated immediately after infection. It acts after some time, okay. This is the MAC complex. You can see all the molecules are there. C5b is there, C7, C8, this all, okay. And molecules of C9. This is the MAC. Okay. Bleb formation is there. You can see the killing of E. coli in the electron microscopic picture. Membranous bleb formations are there, which indicate the killing by complement systems. And here you can see the surface of a RBC membrane. This is the C9 polymer. All the molecules of C9 are there, and this is the surface of a RBC membrane. You can see these holes, these lesions are formed. These lesions are formed membrane of RBC due to membrane attack complex, okay. And classical pathway is activated by, we know this antigen antibody which were activating the classical pathway, IgM and IgG, right? IgA was activating the alternate pathway, okay. Rest all are alternate pathway activating. So IgM is the strongest activator of the classical pathway, and IgG is the second most potent activator. So IgM strongest activator, so requires one molecule only. IgG requires 2 molecules for activation, right? C3a, C4a, C5a, these are known as anaphylatoxins. These all which are living. C3a was being, you can see here. C3a was being C3a was going away, okay. C5a was such like going away, okay. And C4a also, this C4a also, understood? Sorry, C2, what I'm saying, yeah. So C3a, C4a, C5a, these are known as anaphylatoxins. Understood? In classical pathway you must you have to remember this all things, like what is a C5 converter, what is a C3 converter, you should be able to remember. Okay, C3 converter is C1 4B 2A, C1 4B 2A. Okay, 4 is the exception, just remember like this. And here sorry, 2 is the exception. Okay, not 4, 2 is the exception. And C5 converters is C1 4B 2A 3B. Okay, understood? Okay. Yeah, so anaphylaxis toxins have receptor on mast cells and basophils. When this are bound, like C3A, when it gets bound to this receptor for C3A, okay, it will cause degranulation, release of histamines. This histamines granules are released out. Okay, and this mediates the anaphylaxis and inflammation, mediator of anaphylaxis and inflammation. C3b is a opsonin, C5a is the chemotactic agent for neutrophils. Okay, C3b is a powerful opsonin, just remember like this, and C5a is a chemotactic agent for neutrophils. Membrane attack complex, how can you write? C5b, 5b is bound, 6 7 8 9. Okay, just like this line up. Okay, so that completes the classical pathway, and we'll come to the alternate pathway the next. Okay, let's discuss about the alternate pathway now. Alternate pathway is the most abundant complement protein in the serum is C3. Okay, it is a highly unstable due to presence of unstable internal thiouster bond. So C3 when breaks down into C3A and C3B. The C3B is a opsonin, highly reactive. In the absence of any activator also, C3B combines with H2O and gets inactivated. So C3B plus H2O, IC3B, inactivated, right? But most of the mammalian cell membranes have increased concentration of sialic acid, which also inactivates any C3B attaching to the cells. Okay, so alternate pathway is an inducible pathway. C3B is normally in the inactivated state due to combining with water and sialic acid concentration. What are the activators of alternate pathway? Any LPS or endotoxin, any teichoic acid, chitin, all the cell wall things and cell membrane things. Okay, IgA, D, G4 and CRP. Okay, IgA, D, G4 and CRP, these are activating, and snake venoms also. These are the activators of alternate pathway. Very easy to remember. All the cell walls and membrane things you can write. And IgA and D we know from our discussion. IgG4 was the exception. IgG was activating your classical pathway, but IgG4 was the exception, right? And CRP and snake venom, these two you have to remember. What is the mechanism? First, the C3A splits into C3A and C3B. C3B binds to the LPS. Let's say this is a LPS or a fragment of LPS. C3B will bind to this. Or what happens is that C3 will bind and bind and split. Either it splits and binds, or it binds and split and again bind. Okay, attach. So C3b will only bind. This C3b in the presence of factor D, stimulates the factor B, breaks this factor B into BA and BB. This BB binds back to this. This BB will binds back to this. This LPS, C3b and BB. This is known as alternate pathway C3 convertase. Very short half-life, 5 minutes. So this prolonged in the presence of properdin 30 minutes, 5 minutes to 30 minutes, 6 times it is prolonged, right? So LPS, C3b and BB, this is the alternate pathway C3 convertase. C3 convertase means in the presence of this, it will split the C3 into C3A and C3B. C3A will go away, C3B will bind back. Okay, so this is the C5 convertase, as you might have guessed. LPS, C3B, BB and C3B. Understood? Both the two molecules of C3B are there. Okay, step 4, it will cause the binding of C5. C5 will come, C5A will go away, C5B will bind, but not bind. Okay, because this is a catch. C5B never binds. Okay, won't bind back or form MAC, membrane attack complex, with the help of C6, C7, C8 and C9. Same concept. So alternate pathway is activated right after infection, and the same pathway will again form. C6, C7, C8, C9 will form the MAC like this. This same MAC will be there. The MAC complex is always the same, any pathway. This same MAC complex will be there. Same MAC pathway will insert holes and kill the membrane, kill the microbe, right? This is the mechanism of the alternate pathway. So Okay, let's finish the lectin pathway first, and then we'll revise all the three pathways again. Okay, because it is an important part, right? So lectin pathway. Lectins are the proteins which recognize and bind to specific carbohydrate targets. Okay, these are recognizing and binding to specific carbohydrate targets. Okay, so carbohydrate target, the main one is the mannose binding lectin or MBL, mannan binding lectin or mannose binding lectin, MBL. So this is the substrate of the lectin pathway. Okay, so this activates the lectin pathway by binding to mannose present on surface of certain microbes. Okay, so mannose binding lectin is basically a lectin, a family of lectins, okay, which activates the lectin pathway by binding to the mannose present on surface of certain microbes. What microbes? We can remember by CSNL, BSNL, you know, this is CSNL. Okay, you can remember CSNL. Okay, CNL, SL, it is written, no problem. CSNL, you just remember like this. C means Candida albicans and Cryptococcus, which are fungi. S means Salmonella, okay, Salmonella typhi and all. And CS and N means Listeria, Neisseria and Listeria. Okay, MBL is also acute phase reactant in inflammatory response. This is also important. Okay. Function of MBL and lectin pathway is similar to C1q because they are structurally similar. So function of MBL is same with C1q. So what happens? Step 1 is the MBL comes here and the mannose is present upon the microbe. Okay, the microbe is displaying this mannose proudly. This will bind and the microbe starts to cry. Right. MBL recruits two factors, MASP1 and MASP2. MASP means MBL associated serine protease 1 and 2. Okay, so this C1q, this is similar to C1q. I told this MBL is similar to C1q. So this MBL is bound to the MBL, MASP1 is bound, which is similar to C1R, and MASP2 is bound, which is similar to C1S. Okay, QRS, understood? So you can see this is going like the classical pathway only, but the names are different. Like MBL is similar, structurally similar, but they're not the same thing. Then C4 comes. What comes? C4 comes and collides. C4A will go away, C4B will bind. Again C2 come, C2A will bind, C2B leaves. Okay, C2 is the exception, we all know, right? So this is the C3 convertase of lectin pathway. Okay, so C3 convertase will contain what? C3 convertase will contain MBL, then MASP1, MASP2, C4 and C2A, right? So C3 convertase converts C3 into C3A and C3B. C3B will bind back and C4A goes away. C3B binds back like this. Okay, and this part is called the C5 convertase of lectin pathway. C5 convertase of lectin pathway, right? Again, this C5 convertase changes C5 into C5A and C5B. This C5B will again won't bind back, it will form the MAC again, same story. Okay, rest all same, but above two pathways. Lectin pathway can be activated right after the infection. Okay, so alternate and lectin pathway can be activated right after infection, but this classical pathway takes time for activation. Understood? After that, let's go into the disorders of complement system and regulation. Then afterwards we'll repeat the complement systems again. Okay, so what are the disorders of complement system? Deficiency of C1 to 4, 1, C1, C2 and C4 will lead to persistence of immune complex, is called immune complex mediated diseases. Okay, it is autosomal recessive disorder. Deficiency of C1s esterase inhibitor, C1s esterase inhibitor. Okay, C1s esterase is basically an accelerator and C1s esterase inhibitor means a brake. Okay, so this is deficiency, so there is more accelerator, that is C1s is esterified more. Okay, this will lead to unregulated activation of classical pathway, more classical pathway, excessive anaphylatoxins. What are the anaphylatoxins? C3, C4, C5. A. Receptors on mast cell basophil, more histamine means more there may be vasodilation and edema and all such things, laryngeal edema, or it may cause edema of any muscle. Okay, and death. This is called as hereditary angioedema, blood vessel edema. Okay, hereditary angioedema, which is the autosomal dominant condition. Okay, deficiency of C3 proteins will cause autosomal recessive condition. Okay, and because C3b is a very good opsonin, so increase incidence of pyogenic infection. If there is deficiency, there is an increased incidence of pyogenic infection by staphylococcus and streptococcus. Okay, you know about the pyogenic bacteria which are there, I told about you. Then we have the deficiency of late complement proteins, which are C5, C7, C8, C9. So there will be decreased MAC formation, increased incidence of gram-negative infections. Okay, because MAC is basically ineffective in gram-negative. Okay, they are more susceptible for gram-negative only. Okay, then especially Neisseria infection. Okay, incidence by Toxoplasma gondii also. This is autosomal recessive condition. Okay, autosomal recessive. This hereditary angioedema is autosomal dominant. Just remember this, autosomal dominant. Hereditary angioedema, autosomal dominant. Understood? Then another thing, deficiency of C9. Most patients are asymptomatic. Some are clinical features are similar to some this Toxoplasma gondii Neisseria infection may be seen in some. This is also autosomal recessive. Except disorder 2, all are autosomal recessive. Disorder 2, which is hereditary angioedema, laryngeal spasm and all, autosomal dominant, right? Before discussing the regulation, let's move on quickly through the disorders first. Then we'll discuss the regulation. Then again we'll go into the pathways. Okay. If I repeat 2 times, you will remember automatically. Disorders of complement system. What are the disorders? I told deficiency of C1, C2, C3, C4 proteins. First, the persistence of immune complex mediated disease, not much special. Deficiency of C1 as esterase inhibitor, which will cause the break fail. Break fail means unregulated expression, which will generate more anaphylatoxin. More anaphylatoxin means more histamine. More histamine means laryngeal edema. Laryngeal edema means death. This is called as hereditary angioedema, which is the autosomal dominant condition, right? So C1 esterase inhibitor deficiency will cause hereditary angioedema. Then C3 protein deficiency and C3 protein deficiency is there. C6, C5, C6, C7, C8 protein deficiency is there, and C9 protein deficiency is there. So C3 means it's a good option. So more incidence of suppurative infections, okay, and staphylococcus and streptococcus infection. Late complement protein means MAC formation. Think about MAC. MAC formation decreases means more infection by gram-negative, especially Neisseria, okay, and increased infection by Toxoplasma gondii. Then deficiency of C9 deficiency means most patients are asymptomatic, okay, autosomal recessive. This is the fourth factor, okay. Then we'll discuss about regulation of complement system. Regulation is simple. Classical pathway is regulated by antigen antibody complexes and not by antigen antibody alone. Regulated at the level of antibody itself, and some serum proteins also regulated, like C1s esterase inhibitor. Okay. Alternate pathway is regulated by 2 things: factor H and properdin. Factor H and properdin both are important. Okay, and DAF also, decay accelerative factor also. Okay. Factor H holds on to the C3b and forms a complex. This is cleaved by factor 1, which is a protease, and decrease the C5 convertase formation, which is decreasing the C5 convertase formation. Basically, factor H will hold on tightly to the C3b. Okay, hold on tightly to the C3b, and this C3b, if it holds tight, then C3 convertase won't be formed. Ultimately, C5 convertase will not be found very good. Okay, so that is decreased C5 convertase. Then properdin is there. Properdin belongs prolongs T half of alternate pathway, okay, to 30 minutes. We know about this all. In the absence of properdin, C3 convertase is basically 5 minutes, and properdin will prolong it to 30 minutes, right? Protection of human cells from lysis by MAC because of decay accelerating factor. Decay accelerating factor binds to the C3b and C4b and decrease formation of C3 and C5 convertase and decrease the MAC formation. Okay, so human cells are protected from lysis by decay accelerating factor, or CD55. The marker is CD55 cells. Okay, so CD55 is decay accelerating factor, DAF, which is protecting the human cells from lysis by MAC by decreasing formation of C3 and C5 convertase. Okay, let's move on to the pathways very quickly, and then we'll move ahead. So classical pathway, I told classical pathway, first step was the binding of antigen to this site, and CH will open up. CH will cause the attachment of C1q, R and S, C1q, C1R and C1S. C1S is esterase activity. Then after C1, what comes? After C1, which will come? C4 will come. Okay, C1 and C4 will come. C4b will bind, C4a will leave. Okay, after that what will come? 2 will come. 2 is the exception. C4A will bind and C2B will go away, right? And this is the C3 convertase. This is C3 convertase, means 1 4 2. Okay, then what will come? Yes, C3B will come. C3 will come, C3B will bind, C3A will go away, then C5 will come. Okay, means what is happening? Odd, even, then odd, right? Like that. C5A, C5B won't bind, C5A will go away, of course. C5B won't bind, but it will conspire with others like C6, C7, C8, C9 and form a membrane attack complex, or MAC, which will insert holes in the membrane, okay, and kill the microbe. MAC find it difficult to attack gram-positive organism. Then we'll talk about the anaphylatoxins. This is the membrane attack complex, C5B, 6, 7, 8, 9. Alternate pathway, C3 was the main culprit. C3 was there, C3B was there. It will that C3B is bound, okay, after lysis. Then C3B in the presence of factor D. Factor D converts the factor B into factor BA and BB. Okay, BA will go away, BB will bind. So C3B and BB. Okay, C3B, BB. This is the alternate pathway, C3 convertase. Very short pathway, alternate pathway, C3 convertase. So this will cause the lysis of C3. C3 convertase will cause the lysis of C3, obviously. So C3A and C3B. C3B will bind again. C3B will bind again. So C3B, both C3Bs are there and BB is there. Okay, this is called the C5 convertase of alternate pathway, C5 convertase. After step 4, this C5 convertase will cause the lysis of C5A and C5B. C5A will go away. C5B again won't bind back but form MAC. Okay, C6, C7, C8 and C9. Okay, this is the thing. And again the same thing, killing of microbe will occur. Then lectin pathway. Lectin and protein recognized by to specific carbohydrate targets. Okay, MBL is the main lectin, mannose binding lectin, mannan binding lectin. Okay, basic thing is that MBL, the mannose is found in the surface of CSNL. CSNL means what? C is for Cryptococcus and Cryptococcus as well as Candida albicans, right? CS, S is for Salmonella, N is for Neisseria and L is for Listeria, right? Then what is the whole point? MBL will bind first. After MBL, it will recruit MASP1, MASP2. MASP1 is similar to C1R, and MASP2 is similar to C1S, right? MBL is similar to C1Q. After that, 4 will come directly. 4 will come and bind and collide. Then 2, 4. After 4, 2. Okay, same as the classical pathway. So 2 will come, 2A will bind, of course, exception. This is a C3 convertase of lectin pathway. Then C3 will come. C3 will come means this will be the C5 convertase, and C5 convertase, after what changes the C5 into C5A, C5B won't bind back, form the MAC, and again same the killing. Okay, that completes our complement system. Let's move ahead to the next part. Okay, so there is a short part, immune response by T cells. You know about the immune response basically. We are going to a little bit detail. Exogenous protein MSC1 presented to TH cells, okay, and MSC2 presented to the TC cells, this all, okay. And another thing is that MSC molecules are highly polymorphic. We know this, codominant and polymorphic, we know this. So there are some hyperactive mutation sites in the gene encoding HLA-A-LET. Okay, so this is the protein which is encoded by this gene, and here let's say HLA-A21, this is the corresponding part on the MHC molecule. Okay, if there is a mutation in A21 part, there is a mutation in this part is changed. Okay, the mutation will be reflected here. Okay, and this hyperactive part will remain the same. If there is a mutation in this part, basically in this part, this is the mutated part, there is no change here, sorry, there is no change here. Here there is a mistake here. Okay, there is no mutation here, actually there is mutation here. Okay, so mutated part is only mutated. So T cells, TH cells, when get activated, can go into any of the pathways, TH1, TH2 or TH17 pathways, right? So generation of TH2 subset in response to exogenous protein. What is the TH2 subset? How the TH2 subset is generated? Okay, whenever there is a microbe, it is internalized and broken down by the APC, antigen presenting cell. This APC has a MHC2 molecule, and this T0 cell, that is TH0 cell, naive T cell. Okay, it has a TCR, and there is an exogenous protein fragment which is bound. Okay, this is presented to here, understood? And so if it is presented here, there is a signal going inside. Okay, Another thing is that this APC, this antigen presenting cell, it will produce some interleukin 1. It will produce interleukin 1. So interleukin 1 will act on this interleukin receptor present on the TH cells. It will give another signal, second signal. So first signal is by this antigen presentation, second signal is by interleukin 1, third signal B7 is the co-stimulatory molecule present on the antigen presenting cell, dendritic cell specifically. And on the T cell, on the T naught cell, TH naught cell, we have the CD28 molecule. This both are the second co-stimulatory molecule. These also bind to each other and give the third signal. Okay, so we need three signals. Okay, TH naught cell is not inactive cell, basically it is naive because its work was previously not designated. So one of the TH naught cell will come and bind to this part. This is called as clonal selection. Primary stimulation is given by interaction between TCR CD3 complex or TCR T cell receptor complex with the antigen MHC complex. We know this. This is the first signal. This is the first signal, right? Co-stimulation is done by B7 present on antigen presenting cell with CD8 molecule present on TH naught cell. I told you this is the co-stimulation, right? This is the second stimulation, co-stimulation. Okay, interleukin 4, this interleukin 4 with source is unknown. If it is present, it will convert the TH naught cell to TH2 cell, TH naught cell to TH2 cell, and this TH2 cell will be acted upon by interleukin 2. Okay, interleukin 2, and it will multiply. So multiplication of TH2 cell will require interleukin 2. It will require interleukin 2, and if it becomes the TH2 cell, it will produce other interleukins like interleukin 4, 5, 6, 9, 10, 13. Okay, 4, 5, 6, 9 is important, 10, 13 also. Okay. Simultaneously another thing is going on. So what is the other thing which is going on? Other thing is that, look at this. This is a B cell. B cell will have receptor for interleukin 4. Okay, and this is a micro which is bound. If the interleukin 4 gets bind to the B cell, after binding of interleukin 4, there will be proliferation. IL-4 is the B cell growth factor. Okay, there's a growth, sorry, not proliferation, growth. Proliferation is the same concept, but term is different. B cell growth factor, IL-4 is B cell growth factor. So this IL-4, if it's present here, okay, after binding of IL-4, it will cause this proliferated cells. If IL-5 gets bound to the receptor, IL-5 is the B cell differentiation factor. It will get differentiated into plasma cell. This plasma cell will produce antibodies. These antibodies will be similar to the BCRs. Do you remember this property where the antibodies were similar isospecificity? Okay, and this B cell receptor will be similar to the B cell receptor, which has caused the formation of these antibodies, right? So IL-2 produced by the TH1 subset, is growth factor for TH1 cell, TH2 cell, TC cells. Okay, so interleukin 2 is called as the TCGF, T cell growth factor. T cell growth factor, which one is this? Interleukin 2. And this antibody, what can they work? They can cause ADCC, antigen-dependent cell mediated cytotoxicity, opsonization, or classical pathway of complement activation. Okay, and in case significant tissue damage also occurs, this is called as hypersensitivity reaction, which we will discuss the next and the last, right? Also there is a CD3 present over here, and the CD4 molecule is there. CD4, you know, is the pan T cell marker. Okay, this will touch this MHC2 to make sure that this is the antigen presenting cell or not. Okay, this is another signal, right? So this is the whole immune response by T cells, how they are mediated. Understood? Let's move on to the next part. Okay, so T helper cells get activated with 3 pathways basically. You can we told that 2 pathways, but another pathway is there, TH17. TH1 pathway is seen in the granuloma formation. TH2 pathway is basically in the allergic responses. TH17 pathway is basically activated in any disease with acute on chronic inflammation. There is a phenomenon called as acute on chronic inflammation. TH17 pathway is seen there. Okay, so TH1, 2 and the 17 pathways. Okay, let's discuss the hypersensitivity reactions. So hypersensitivity reactions are of 4 types basically. 4 types of hypersensitivity reactions are there. Type 1 is think of allergy. Type 1 means think of allergy first. Okay, type 2, think of autoantibody formation. Autoantibody, allergy, autoantibody. Type 3, think of immune complex circulation of immune complex. Okay. Type 4, think of cell mediated immunity. So type 1, think of allergy. Type 2, think of autoantibodies. Type 3, think of immune complex circulation. Type 4, think of cell mediated immunity. Right? Correct? Type 1, hypersensitivity reaction. All allergies are mediated by type 1 hypersensitivity reaction. Very important point. So here is a typical allergic response. Here let's say this is the time axis, this is the response axis, this is the response, allergic response, okay, reaction, and this is the time. So basically when there is exposure, 2 hours the immediate reaction. Immediate reaction lasts for 2 hours like this. There's a high peak, and after this there's a lag period here, and after 4 to 8 hours there's a late phase response also. There's a late phase response, okay, and last up to 1 day. It is a typical allergic response. Yeah, so how this is mediated? First exposure, let's say a allergen came, a pollen grain for example. There is a time lag first. This time lag is called the sensitization period, okay, and after the second exposure the reaction starts. Okay, there is a sensitization period or the time lag between the first exposure of allergen and the second exposure of allergen. This time lag is called the sensitization. Okay, and yeah, so what is the mechanism? Let's say pollen grains are inhaled. So macrophages in the airways, dendritic cell, will engulf them. This dendritic macrophage engulf the pollen grain. It will dendritic macrophage means it will what it is a antigen presenting cell, APC. Okay, this dendritic macrophage obviously is a APC, antigen presenting cell. It will present it to the T cell. This T cell will bind to the allergen and activate the Th2 pathway. Th2 pathway will cause release of IL-4, 5 and 13. 4, 5 and 13. Okay, I am just telling you those which are important. Okay, because 4, 5, 6, 10, 13 all are there. Okay, 4, 5, 6, 10, 13 all are there, but we are taking 3 of them, 4, 5 and 13. Okay, 4, 5 and 13. IL-4 is a B cell attractant. Okay, it will stimulate the B cells. We read about the type. IL-4 is the B cell growth factor, so it will produce the IgE antibodies. Okay, B cell stimulate the B cell and produce the IgE antibody. Interleukin 5 is a chemo that attractant. Okay, it stimulates the eosinophils, increase the eosinophil. The IL-5 increase the eosinophil levels, right? And IL-13 increase the IgE production and increase the secretions, increase mucus secretions basically, and IgE production also is increased. So stimulate the B cells. B cell will produce the IgE. This IgE antibodies will bind to the Fc receptor on the mast cell. Okay, it's IgE. What it will do? IgE will bind to the Fc receptor on the mast cell. Okay, so let's say this is a mast cell. The Fc receptor is there. Here IgE is binding. Okay, done. This is called as sensitization. Okay, this is called as sensitization. So this is the first time exposure, but when there is a second time exposure, okay, this after sensitization it is dormant. When there is a second time exposure, the pollen grains again get bind to the IgE. This binding will cause the activation of mast cells now. Okay, so first exposure is well and fine, no much response. Okay, but second time exposure, where there is second exposure of pollen grain, these are getting bound, and binding causes activation of mast cells. When their mast cells are activated, they release preformed mediators and newly synthesized mediators. Okay, preformed and newly synthesized, many mediators are there. So basically discussing the mediators, preformed mediators means within minutes there will be reaction. Preformed mediators, for example, histamine, enzymes, proteoglycans and phospholipid A2. Okay, proteoglycans, for example, phospholipid A2. Understood? So preformed mediators means histamine will cause vasodilation, it will cause congestion in nose, it will increase the mucus secretion. Enzymes, protease enzyme, tissue damage, cause release of phospholipids and attract inflammatory cells, right? Inflammation will be started. Proteoglycans will be released. Proteoglycans, what they will do? They will increase the size of tissue and make it gel-like. Proteoglycans make the tissues gel-like. Okay, like phospholipase A2. Okay, phospholipase A2. It will stimulate prostaglandin D2 and leukotriene B4. Prostaglandin D2 and leukotriene B4 are powerful chemotactic agents. Okay, and also produce leukotriene C4, D4, E4, cause vasodilation and bronchospasm. Okay, vasodilation and bronchospasm. Basically all are causing like symptoms like this, vasodilation and bronchospasm. Thus anaphylactic reactions are highly dangerous reactions, right? And newly synthesized mediators constitute the late phase reactants. These are the early phase reactants, these are the late phase, 2 to 24 hours, right? And they are mediated by eosinophils. Eosinophils are degranulated, produce galactin 10. Okay, in case of asthma, you must have heard in pathology, your charcot laden crystals are seen in the sputum in asthma, charcot laden crystals, and these are the evidence of degranulated eosinophils. Okay, so increased cytokines, increased cells, that galactin 10 will cause the increased cytokines, will increase the cytokines, increase the number of cells. There is a vicious cycle, again there is anaphylaxis, and this reaction goes on. Okay, so allergen, sensitization, second exposure, degranulation, then will cause immediate reaction and late reaction, and again all the mediators and all. Okay, so hygiene hypothesis, this is often asked, that environmental factors have more risk to cause allergy. Hygiene hypothesis is that due to more socioeconomic status, the immune responses have become, allergies are more common now in developed nations. Okay, extremes of temperature, nuts, milk, all of this can cause allergy. Okay, we are finished with type 1. Let's talk about type 2 hypersensitivity reaction. Type 2 is the antibody mediated hypersensitivity. Okay, antibody mediated. So antibody production after it is produced can be can work by three ways. Antigen antibody reaction will be there. It can be eaten up by macrophages, or it can activate complements, cause inflammation, right? Complement means killing an inflammation, or can destroy the antigen completely, right? So first mechanism, what's the opsonization and phagocytosis? When antibody is produced against cell, the entire cell can be engulfed. Okay, like AIHA, autoimmune hemolytic anemia, antibody is produced against the RH antigen. Okay, all the cells are engulfed, all RBCs are engulfed. ITP is immune thrombocytic purpura, where the platelets have an antigen GP1B9, GP1B9, and the antibodies are produced. This go to spleen, and they are engulfed by the splenic macrophages called the littoral cells. Okay, this is the fact. Or they can complement an Fc mediated inflammation, Fc receptor mediated inflammation. Okay, like membranes and skin cannot be eaten by macrophages by phagocytosis, but pemphigus group of disorders is the example where antibodies are produced against the desmosomes and hemidesmosomes. Okay, this desmosomes and hemidesmosome, antibody are produced against them. So antibody bind to the antigen on the surface of the skin, activate complements, inflammation, destroy the antigen, result in swelling and blister formation. Example, glomerulonephritis, antibody against collagen fibers. So this will cause some complement activation and destruction. Then the third type is antibody mediated cellular dysfunction. Okay, type 5, basically in older tests, type 5 hypersensitivity reaction is also given. This is now classified into type 2 HS only. Okay, type 2 hypersensitivity only. So antibody will directly bind to the receptor and destroy the receptor. Antibody will directly bind and destroy the receptor here. Like myasthenia gravis, antibody produced against ACH receptor, directly destroyed. Graves disease against TSH receptor, activation of TSH, growth of thyroid, goiter will be there, and all. Then intrinsic factor produced by parietal cells, if this decreases, decreased absorption of vitamin B12, pernicious anemia, right? These are the examples. You must know the examples of this type. These hypersensitivity reactions are more important from pathological point of view rather than the microbiological point of view. Okay, so I have given some pathological examples which you can use to correlate it very clinically. Okay, clinical correlation is important in this case. Okay, after that, type 3 hypersensitivity reactions. Type 2 is done. Type 3 is SLE, PAN, PSGN, and reactive arthritis. These are all the prototype disorders, okay, of type 3. In these disorders, type 3 hypersensitivity occurs. Okay, how it occurs, we'll discuss it, and how to diagnose what is Arthur's reaction, and lastly we'll discuss the type 4 hypersensitivity and finish up the immunology topics. Okay, yeah. So SLE is the systemic lupus erythematosus, PAN is polyarthritis nodosa, PSGN is post streptococcal glomerulonephritis, RA is reactive arthritis. Okay. Pathogenesis. What is the pathogenesis of type 3? Type 3, what happens is the formation of immune complex any foreign antigen is induced, which will cause the formation of antigen plus antibody form a immune complex in circulation, inactivated immune complex. Then deposition of immune complex. This immune complex get deposited on the endothelial surface. This get deposited on the endothelial surface. This deposition can cause fibrinoid necrosis. This you must have read in pathology, or if I haven't read, you can ignore this. It can cause thrombus formation. Thrombus, you know, a clot type of formation will be there. Thrombus formation. They can activate the complements also, C5 and C3, which can cause inflammation and vasculitis, right? And inflammation and tissue damage. We can cause this. Okay, basic easy thing. Okay, any foreign antigen introduced, antigen antibody formation, immune complex is formed, so move into circulation. Then they are deposited in deposition of immune complex occurs. When this is not deposited, can cause fibrinoid necrosis, or can cause thrombus formation, or can activate complements like C5, C3, which can cause inflammation and vasculitis. How to diagnose type 3 hypersensitivity? Fibrinoid necrosis, decreased serum complement levels, decreased serum C3 levels, right? This diagram is important pathological point of view. You can ignore this. Arthur's reaction is a localized serum sickness to explain type 3 hypersensitivity. Basically, skin test. Now it is not done. Okay, Arthur's reaction. Okay, then let's come to type 4 hypersensitivity reaction. CD4, you know, that marker is CD4, T cell marker, and T cell may be of two subtypes, three subtypes basically, TH1 and TH2, TH17, TH1 mediated, TH17 mediated. If you see in the microscopy, TH1 mediated, you will see granuloma formation. TH17 mediated, you can see acute and chronic inflammation. Okay, rarely you can have CD8 mediated type 4 hypersensitivity. Type 4 hypersensitivity is a characteristic of CD4, okay, hypersensitivity, but we can have also CD8 mediated type 4 hypersensitivity. Look at this. Look at this, what is happening basically in this part, and we'll discuss a combination also. We'll discuss a combination of CD1 and TH1, TH2 pathway. So here the pathogenesis is basically we're talking about TH1 pathway. Okay, so APC is there. It will present let's say tuberculosis. It is presenting the tuberculosis antigen. Okay, this is recognized by the CD4 cells, because antigen presenting cells, MHC2, right? This is the MHC2. Okay, TH1 pathway is activated. TH1 pathway is caused produce INF gamma only. Okay, interferon gamma. This interferon gamma will convert the normal macrophage into M1 activated macrophage. Interferon gamma has the function of converting normal macrophage into M1 activated macrophage. Okay, this M1 activated macrophage will produce IL1 and IL12. IL1 and IL12 will attract the lymphocytes and form the granuloma. Okay, understood? So a normal macrophage is being converted to M1 activated. M1 activated is causing the formation of interleukin 1 and 12, which are attracting lymphocytes and causing the formation of granuloma. So can I give example of Mantoux test for TB? This is important in the case of microbiological point of view. Mantoux test for TB and contact dermatitis. Okay, so contact dermatitis not type 1, this type 4, and this follow the TH1 pathway. TH17 pathway is seen in two conditions, PU, you can remember, UP, Uttar Pradesh, ulcerative colitis and psoriasis, you can remember this. Predominantly neutrophils and lymphocytes are seen in microscopy. Acute and chronic inflammation told us. TH17 means acute and chronic inflammation. Acute inflammation means neutrophil, chronic means lymphocyte, right? Combination of TH1 and TH17 is seen in rheumatoid arthritis and multiple sclerosis, RM. Okay, rheumatoid arthritis and multiple sclerosis will show a combination of TH1 and TH2. Understood? Okay, CD8 mediated type 4 hypersensitivity. This is a rare reaction, but we are discussing it here. CD8 mediated type 4 hypersensitivity. How CD8 is mediating hypersensitivity? CD8 T lymphocyte is also known as CTL. CTL means cytotoxic T lymphocytes. Okay, CD8 T lymphocyte are also called as CTL. CD8, when they are activated, they release perforins and granzymes. So in viral hepatitis, in case of viral hepatitis, there is CD8 mediated damage, means there is perforin create pores, and the granzymes will go on and cause the function. These granzymes will function like caspases. Okay, this is the thing. Understood? So type 1 diabetes mellitus also we have CD8 associated damage. CD8 consider insulin and glutamic acid foreign in predisposed individuals. Okay, so if individual is diabetic, means there is some genetic defect in this individual. Type 1 is genetic, right? So CD8 will consider insulin and glutamic acid, GAD, glutamic acid decarboxylase enzyme. Okay, this will foreign, this is recognized as foreign, this both. Okay, so it will activate the CD8 cells, it will destroy the pancreatic beta cells, decrease insulin production and cause type 1 diabetes mellitus. So B cell also considered the foreign, so antibodies will be produced against them. The anti-GAD antibodies are diagnostic for diabetes mellitus. You must have known this, or you should remember this part. Okay, so that completes the hypersensitivity reactions. Let's move on to the next. Talking briefly about autoimmune disorders, not much is important. I will read in pathology as well. Tolerance. So what is tolerance? Tolerance is the ability, tolerance you must know from microbiology, ability of lymphocytes to identify self and non-self is the tolerance. So you have central tolerance and peripheral tolerance. Two types of tolerance are there. Central tolerance, whichever is escaping the central tolerance, is taken care of by peripheral tolerance. Central tolerance, we have thymus, organ of reticular endothelial system. Thymus, we have macrophages. During embryology, whenever there is an APC antigen presenting cell or macrophage, this is presenting a skin antigen, let's say, and this is the TCR. So the AIRE gene, okay, autoimmune gene regulator, it is deleting the T lymphocyte, is bind to the self antigen, and whenever there is mutation, will cause autoimmune disorder called polyglandular syndrome. Okay, so AIRE mutation causes polyglandular syndrome. This is the entrance question, and so basically we have tolerance for this region only. Okay, and IgM and IgD receptor is there, you know, on the B cells, and variable region BD1J1 combination. If it binds to the self antigen, either it dies by apoptosis or it dies by receptor editing. Okay, so newly formed B cells will never bind again because receptor is edited now, it can't bind. So self antigens are protected from the own immune cells by this process, by tolerance. But if some escape the central tolerance, they will be taken care of by peripheral tolerance. If something misses central tolerance, it will be taken care of by the peripheral tolerance, right? First thing is the energy. Okay, energy. What is energy? Energy is if you bite your tongue, okay, tongue antigen goes into circulation, APC will be digesting it, okay, MHC2 will present it. This is a tongue antigen TA, which is escaped your central tolerance, let's say. This is presented to CD4 cells, and any CD4 cells bind to self antigen, there is the production of CTLA4 and PD1. CTLA4 and PD1. Just remember this two molecules, CTLA4 and PD1. The CTLA4 and PD1 destroy CD80, CD86 and CD28. Okay, basically what is happening, antigen presenting cell MHC2 and T cell TCR. Okay, these are the things. A co-stimulatory molecule is there in the antigen presenting cell, CD80, CD86. Okay, and the T cells we have the CD28. These both are co-stimulatory molecules, but this CTLA4 and this PD1 will destroy this co-stimulatory molecules, which are giving the second signal for T cell activation. So if co-stimulatory molecules are destroyed, T cell is not activated. Now if T cell is not activated, you will be not having any, the body will not mount any immune response against the tongue antigen, right? Next for peripheral tolerance, another mechanism is by T regulatory cells. Okay, these are the mechanisms. One is the energy, another one is the Treg cells. Okay, so T regulatory cells have markers like CD25, FOXP3 and alpha chain interleukin 2 receptor. These are this type of markers. Okay, so whenever there is a antigen bound to the T regulatory cells, it gets suppressed. Like if mother and against the placental antigen, okay, placenta possesses extremely high Treg cells, okay, which will suppress the Hofbauer cells, the placental macrophages. So fetus is not rejected by the mother. Okay, placenta is foreign to the fetus, na? So but this placenta has Treg cells. Treg cells have a ability to suppress the macrophages. Treg cells can suppress the macrophages and the fetus is not rejected. If FOXP3 is mutated, the marker, this FOXP3, this is mutated, will suffer from syndrome like IPEX syndrome. IPEX stand for immune dysregulation, polyendocrinopathy, enteropathy, X-linked syndrome. Okay, immune dysregulation, polyendocrinopathy, enteropathy, X-linked syndrome. And mutated CD5 or interleukin receptor will cause multiple sclerosis. Then we have third mechanism, which is a fast fall ligand, apoptosis mechanisms. Okay, apoptosis, you know about the fast ligand. Fast ligand is called the death ligand, and fast cell receptor, you know about that. We discussed this. This was also a type of your ADCC, antigen antibody detector cell mediated cytotoxicity. We discussed it earlier, right? Talking about immunologically privileged site, this is the fourth mechanism. This is the fourth one. Okay, so testis, brain, and uveal tract. Testis, we have blood testis barrier. Brain, we have blood brain barrier. Okay, which are not exposed in utero to the thymus. Okay, so if one side testis is destroyed by trauma, the other testis will also have a chance of destruction by immunity, because if one testis is destroyed, okay, the antigens, the testicular antigens which are previously not expressed, which are previously not seen by the body will see it, body will think it foreign. Okay, so traumatized testes have to be removed immediately. Okay, if one testis is destroyed, the testicular antigen will be exposed foreign to the body, so traumatized, any testis which has suffered trauma must be removed immediately as fast as possible. Okay, otherwise there will be destruction of the second testis as well. Okay, and the destruction will be more severe than the first testis. Autoimmune disorder have pathogenic models instead of pathogenesis. They don't have any pathogenesis, they have a model. Okay, many factors are affecting this. Basic introduction. So genetic factors: tolerance, defective tolerance, abnormal display of self-antigens, and infections. Genetic, defective tolerance, abnormal display of self-antigens, infections are basically causing the problem. Okay. Okay, so what are the genetic factors? HLA related and non-HLA related. HLA related, let's discuss some HLA related factors, and some non-HLA will discuss about that. So if HLA B27, if this is 100 present, okay, it will cause ankylosing spondylitis. Okay, ankylosing spondylitis. And HLA DQ and HLA DQ2 and DQ8 are causing celiac disease, which is gluten specific enteropathy. Okay, Indians have very less incidence of celiac disease, since we must have chapatis in childhood. Okay, chapatis are gluten rich, that's why Indians have a little chances of celiac disease, right? So B27 ankylosing spondylitis, DQ2 DQ8 celiac disease. Non-HLA, we have PTPN22, pre-tyrosine phosphatases. Pre-tyrosine phosphatases which destroy the tyrosine kinase. PT22 loss will cause overactivation, increased T cells, and autoimmune reaction. CD25 IL-2 receptor destruction will cause multiple sclerosis. CTLA4 polymorphism will cause type 2 diabetes mellitus and rheumatoid arthritis, right? Environmental factors are you can remember by HIV and G. Okay, HIV gene, you can remember by this HIV gene. Okay, just a silly mnemonic. H for hygiene hypothesis is responsible. Exposure to pathogens in early life will weaken the immune system and increase risk of autoimmune disorders. Okay, then I means infection of streptococci. All trigger molecular mimicry. Example, rheumatic heart disease. Remember we talked about rheumatic heart disease and molecular mimicry sometimes earlier today. And viral infections can predispose to a new antigen. Okay, then gender, more risk is of women. Women have more risk of this autoimmune disorders than men. Okay, due to estrogen as well as hygiene hypothesis. And SLE is the prototype of autoimmune disorders. Okay, so HLE is basically will be dealing with HLE in not in your microbiology. We will be dealing it in your pathology. That's why I am leaving this. Okay, so that completes your immunity part, immunology, whole immunology. It is a 5-hour lecture, so in 5 hours we completed all the aspects of immunology which are there in the microbiology as well in the biochemistry as well. I covered all of them basically. So that's all. I hope you find the lecture very interesting, and immunological mechanisms you have to remember and retain as well as understand very carefully. So do read. If you want to read any textbooks of immunology, you can read QB's Immunology. Okay, you can read QB's Immunology, and Levinson is a very good book, Levinson. And from microbiology point of view, your Anand Narayan and Apurva Sastry. Anand Narayan is a bit old, outdated. Apurva Sastry is good. Okay, and many books are there for immunology, or you can follow Themes Medical Microbiology. In that also immunity is very good, very nicely given. I have taken references from the Lippincott textbook of immunology and QB's textbook of immunology, as well as from your Apurva Sastry. And that's all in today's lecture. I hope I covered all the aspects, and you understood each and every concept. Okay, if you have any doubt, you can contact me at Instagram. This is my Instagram ID, Biswanath.Tripathy _ okay, this is my Instagram ID. You can contact me for any doubts. So keep studying, and do comment on which other parts you want any lecture. Thank you, and have a great day ahead. Good night.